I
So I put up the slides that you sent me.
Good, perfect. We're in
And
good shape.
okay, and if you just tell me when to change them, I'll change them.
Yeah, the problem is from my side, I can't see anything just yet.
Oh, okay.
There we go. There was some software that had to be downloaded to make my computer work with this camera. So I think we're good now. Next slide. Oh dear. Okay, so you guys, I can't ask the class questions. So we did this already though. You guys already heard the whole Chief Seattle story,
Yes.
right?
Yes.
Yeah, okay. So this is a slightly different film thing that PowerPoint presentation than we normally do. Next slide. Okay, so if you guys will recall, so we're doing plant pathology today. And if you recall, we had, when we talked about IPM earlier, we were talking about two of the things that, sort of matter in terms of our day-to-day existence, honestly. Plant pathology is part of, when we talked about integrated pest management, one way we deal with that is the pathogens, all the different diseases that attack things. And when we start talking about specific diseases that affect us in everyday life, we have things along the line. We had pestilence. So, and I think we might have covered this last time, that next slide, ergot causes gangrenous ergotism, and it also is what taught us how to make LSD, and that people spent whole winters tripping and having their extremities fall off at the same time, which was what gave rise to all kinds of fun stuff like witchcraft, and at least gave rise to the firm belief in witches, because people did not understand what was going on with them. That said, that's a pretty dramatic example, but famine has occurred over and over and over again. The Irish potato famine is perhaps the most recent and one of the more devastating ones that we've that's been recorded in history, but famine is happening all across the globe even today, and plant pathogens have a big thing to do with that. Wheat rust, I was just watching on the American Phytopathological Society has a whole thing on wheat rust. Really interesting stuff. It starts in Central America and blows up all the way through the United States into Canada, and we have problems with wheat rust every year, and it threatens repeatedly to wipe out, the global wheat production, and it can go 10,000 miles. One of the more impressive stints was that wheat rust from South Africa traveled all the way to Australia on the wind, and then infected crops there. So it's challenging from a global perspective, and they're always the most effective way to deal with this is to breed resistant varieties of wheat and try to stay, but we're always staying sort of one step ahead of the wheat pathogens. And so this probably harkens back to a lot of what we're all experiencing, on a day-to-day basis right now. The science generally tends to only be 1 or 2 steps ahead of the flu or coronavirus or wheat viruses. And we can die from coronaviruses. We can die from, in the United States, we're not at great risk, but people have historically died when their wheat crops have failed. And that's likely to happen again in the not too distant future. It's not necessarily going to happen in the United States, but in places like Kazakhstan, where they grow wheat and they depend on their wheat crop, they don't have the money necessarily to just import a whole bunch of corn if the wheat crop fails. So, this matters to us on a day-to-day basis. Next slide, please. So, we have talked a little bit about dead white guys already. Anton de Berry correctly identified Phytophthora infestans as causing the Irish potato famine. He's also give germ theory a big boost. He was working in the 1860s and 1870s. Pasteur really put, the final touches on all of this and really proved, but he did that in the early part of the 1900s, just over 100 years ago, which is pretty amazing that, we moved from evil spirits causing plant maladies to pathogens causing maladies. And we like to think that we've been advanced for a long time, and we haven't. There are people who still curse Anton de Berry today because he's responsible for the spelling of Phytophthora and a few other things. But, he did the world more good than harm. And, just because he pisses off a few Latin scholars, I'm going to let him, I'll let him go for that one. Okay, so that's, next slide, please. That's a little bit about, at least the background of plant pathology. And yeah, we're skipping over an awful lot. Other stuff includes Bordeaux mixture on grapes, which is perhaps something that would be worth mentioning here. It's copper lime sulfate, and they spray it on grapes. And they originally did it to keep the school kids from eating the grapes on their way home from school when they'd walk through the vineyards in France. But it turned out that it knocks down a whole bunch of diseases and insects as well. And then it biodegrades, at least to some degree. The copper isn't so, hundreds of years of copper use doesn't do us any favors. But other than that, there's a lot of interesting stuff in terms of the history of plant pathology. You could probably do a whole course on it. So what is plant pathology? It is what the study of medicine is for humans, at least loosely speaking. We're really studying the microorganisms and the environmental factors that cause diseases in plants. So, it's not that far from studying COVID-19, we're doing it in plants, not in people. And they're the mechanisms by which these factors induce disease and the methods of controlling or preventing the disease. If you don't understand the methods by which the factors induce the disease, you're going to have a hard time controlling it. So that's essentially what it is. It's plant medicine, mostly focused on bacteria and fungi. Next slide. And like anything that's a science, the scientists develop shorthand for talking to one another, which unfortunately becomes jargon and then makes it pretty difficult to understand for anybody from outside. So that's one of the reasons I ended up going into this whole mess, is because when I was working as a consulting arborist, I didn't know the difference of a Parathecium from a Sporangium, and on and on and on. And there's all these different terms. And so we're going to go through some of those terms because it's really hard to talk about plant pathology without them. And so I'm not going to introduce them all at once because it gets really hard to remember them all just in little dribs and drabs throughout the talk. So to start with, we're going to talk about, I'm going to introduce the term hyphae or hypha and mycelium or mycelia. I'm doing these backwards, I apologize. So really hypha or is one of these little tiny strands that you see in this image right here. So this is a Phytophthora that you're looking at in an agar media. So think of it like Jello, and it's growing in this Jello, and we can take a picture of it because it's fairly clear. So hypha is singular, hyphae are what we're looking at. So this is a little bit like talking about a twig or a branch. A mycelium, or plural mycelia, are twigs or branches. If you will, it's talking about the mass of twigs or branches. It's a little bit like talking about a tree's canopy or something along those lines. So this is the mycelium. It is the vegetative part of the fungus, if you will. Vegetation is obviously used very loosely because it's not a vegetable, it's not even a plant. And it's what feeds and grows through things. It's not the reproductive structures. Sporangium, next slide, please. I think we've got a slide for sporangium. Oh, okay, so here, this you could look at. This is a mycelium. This is a picture of a fungus, a true fungus in water. So it's just sort of floating there and it's colonized something. I can't even tell you what, probably a seed or something. And so you're looking at the whole mycelium there. You sometimes hear the term thallus. We're not going to use it, but this is opposed to looking at the crown of a tree. You're talking about the whole tree. So when you're talking about a thallus, you're talking about stuff you can't see and including reproductive structures. Mycelium's non-reproductive structures. Don't worry too much about that. We're not using the term thallus. When we're talking about mycelium or hypha, we're just talking about certain parts of a fungus. A sporangium, on the other hand, next slide, is the structure that produces it's a reproductive structure. So you can sort of think of the sporangium as being like a fruit on a plant. Sporangium includes seeds, but the seeds would be the spores. So the sporangium is the thing that includes the seeds and can release the spores. And that's what we're looking at here. Again, this is a Phytophthora, and each one of those sacs that you're looking at is filled with what looks from here to be like hundreds, but it's probably only 20 to 30 spores on the inside. And these are zoospores. Now, these are some of the places where fungi really differ from plants, because the spores that fungi produce are really broadly, they're very different. Seeds of plants tend to be pretty uniform. They've got a hard coat. They can vary from the size of a speck of dust all the way up to, avocado pits, that's a pretty broad range. And the fungi have a broad range too, but where the fungi get different and really strange is the spores they release. Some of them can swim, some of them hunt actively. So they're a lot more like animals than the seeds of plants are. In other instances, however, they're very similar to seeds. So they can produce a whole bunch. So there's different names of spores. You'll hear zoospores, you'll hear chlamydospores, you'll hear oospores, you'll hear all these different terms. Suffice it to say, the only ones that swim and hunt are zoospores, zoo meaning like animal. And then the rest of the spores are all pretty passive in one way or another. A sporangia can act like a spore, a little bit like one might think of an apricot can act like a seed. The apricot isn't really acting like a seed. The sporangium, though, can land on the ground, and if you let it rot there for long enough, it'll sprout a tree. So you can think of the same thing, a sporangium. If the conditions are right, the sporangium doesn't have to release spores. It can germinate directly out and create a new fungal body, a new fungal thallus. Okay, so the spores gets that's the difference between sporangium and spore. Sporangium produces the spores. The spores would actually grow out, although fungi being as plastic in the original sense of the term, meaning flexible, malleable, not really fixed in a lot of things. The sporangium, unlike fruit of a plant, can germinate directly. Next slide, please. So here is a sporangium that has released a whole bunch of zoospores. These zoospores now, they're pretty impressive for a single cell. You can think of them kind of like a sperm with antennae, but they're even more remarkable than that because they've got these single cells have more senses than we do in some ways of thinking about it. They've got a sense of light, they've got a sense of touch, they've got a sense of smell, they've got a sense of gravity. But they also have a few things that we don't have. They can follow electrical gradients. In fact, if you look at the zoospores for Phytophthora soji, which is something that specializes in soybeans, if you put electrodes in the water that exactly mimic the conductance of soybean roots, it'll swim to them. If you put electrodes in water that are lower voltage than soybean roots, the zoospores swim away, or higher voltage, they swim away. It doesn't matter. They can sense electricity and they're attracted to the electrical signature of soybean roots. So I just think that's fascinating. And I keep trying to switch the slides on my own. I'm sorry, Susan. So let's, next slide. Okay, so those are some of the terms. And mostly we focused on fungi because fungi and the things that look like fungi are the things that we're really going to run across 90% of the time. Okay, maybe not 90% of the time. If we include abiotic problems, actually, we're going to run into those more than 50% of the time. But when we're talking about biotic organisms, fungi are by far the predominant pathogens of plants. That's unlike people. People get viruses and bacteria. Fungi, we get very few fungi that attack us, you know, athlete's foot and a few other things. But in the plant world, fungi are the big killers. Okay, so we have, when we're looking at fungi, we've already covered hyphae and sporangia. This is what we normally think of, though, when we see fungi and we think of reproductive structures. Mushrooms. So this is armillaria at the base of a cherry tree, and this armillaria is killing this cherry tree. I mean, you can't see that, obviously, because the cherry tree's, you know, canopy is declining, but it's not in the picture. But this is armillaria, and these are all reproductive structures. So a lot of people will say, oh, you know, I removed all the mushrooms, so I took care of the problem. That's like saying you cut down an apple tree by picking all the apples. If you remove these mushrooms, the fungus that is attacking the tree is still, it's in the roots of the tree and it's in the ground, and it's fine if you pull all these mushrooms out. So the only thing you're seeing half the time when you're looking at mushrooms are the reproductive structures of something that's much larger and living underground. Okay, now, and so most of the time, that's all we ever see of them, because unless you're going to skin the plant, next slide, if you take the bark off that cherry tree, you might see something that looks like this. These are the hyphae. These are all those little mycelia that are growing underneath the bark, and these are mycelial mats that you're looking at. And armillaria produces these beautiful white mats that basically eat the phloem of the plant that it's attacking and kill all the living tissues there and turn it all into fungus. Yeah, okay, so next slide. Okay, so that's fungi very, very briefly. They're these filamentous organisms that live primarily in the soil, sometimes on plants and that kind of thing. What we're looking at here is a bacterium. And if you guys know anything about bacteria and you've been, you know, especially if you're, you know, got a background in biology, you're going to laugh because this is the ultimate bacteria. It has every feature ever seen on any bacteria, right? So you're not going to find a bacteria necessarily that looks like this, but you may find bacteria that have a flagella, which is a long piece that sticks out at the back of some of the bacteria, and they twirl it around and it allows them to, or whip it back and forth, and it allows them to swim through water or other, you know, fluids. Pili, which can allow them to fasten themselves to these are these little short hairs that stick out all over the place. They're often used to stick themselves onto things, so they'll grab onto things. Sometimes they can be used to help it move over things, where it grabs onto some parts and lets go of others, and it can move across a surface that way. And so there's a bunch of different things on here that, you know, are features that you might find on a bacterium. What you will always find in a bacterium, though, is it always has this nucleoid, and it's not really a nucleus, because you'll notice it's not in its own distinct organelle. It's not bound in a membrane or anything. It's just sort of wadded up and stuffed in the middle of this little tiny rod or sphere or whatever shape you've got for your bacteria. And they almost all have some sort of membrane. What they don't all have is a cell wall. So some bacteria have cell walls, and that's what things like penicillin works against, it's a cell wall inhibitor. So it inhibits any bacteria that has a cell wall. It finds it really hard to grow in the presence of penicillin. But there are plenty of bacteria that don't have cell walls. They have membranes, but the cell wall isn't there. And that means that these other bacteria aren't as tough, but they're also harder to mess with in some ways. And you have other things like ribosomes and things and cytoplasm that are in every cell in the world. And that's no different from bacteria to you and me. We have some of these structures in us too. So bacteria tend to be free living. So that whereas fungi tend to be these things that have these big structures that you can see, bacteria are for the most part completely microscopic. There are some colonial bacteria. They're called actinomycetes, and they are important to us in certain regards. And so you will run across bacterial colonies that you can see, but for that's kind of unusual. That's the exception rather than the rule. Okay, next slide. So this should be a slide of actinomycetes. So this is what it looks like under a microscope. Again, now you get enough of this stuff together and you can actually see it with your eye. But this looks like a fungus, and yet if you look carefully at it, you'll see it's not made up of cells that are long and thin that stretch out. This is made up, they're sort of like, if you will, sort of like sponges are to the eukaryotes. Every little tiny bacteria is cemented next to the one next to it in a line, and you can see the bacteria in this image, and they're just they're little rods or little spheres. Okay, next slide. So this is the kind of thing that bacteria typically cause in terms of plant symptoms. We don't see any fungal structures like mushrooms with bacteria. All you get are your classic sort of wilts. That could be caused by a fungus too. This could be monolinea. It's not. This happens to be fire blight I believe this is apple. And fire blight on apple is caused by a bacteria that gets into the flowers, and then it gets into the phloem and kills the plant down to a certain point, and the plant usually stops that at a node. And depending upon the plant species, it's better or worse at it. Asian pears are terrible at stopping fire blight. Pears in general are terrible at stopping fire blight. Apples are pretty good at it. And crab apples are really good at it. Okay, so that's one bacteria. There's 100 others. One of the more famous ones is Pseudomonas. This actually looks mostly like frost damage, and that's because it makes its living by causing frost damage. We'll get into that a little bit later. Okay, next slide. So these are viruses are microscopic. There are no forms, even the exceptions of viruses, that we can see. And for the most part, there's nothing we can, while we can treat fungi and we can treat bacteria, at least within some reason, pretty much for viruses, there's nothing we can do. The good thing about viruses is most of the time when we get viruses, they're not causing enough damage to a plant that we have to worry about it all that much. There are some exceptions to that, but plant viruses generally are incurable. The only way to cure them is to rip the plant out. If the plant is an ornamental, sometimes the viruses make the plant even prettier. I mean, if you read, what do we call it? Botany of Desire, you guys all remember the tulip craze? I mean, you guys all, right? Yes? Okay, yeah. Linda's nodding her head anyway. So viruses, for the most part, nothing to do. Sometimes they can even make a plant look kind of cool. For crops, they're usually just the infected plants are just destroyed, and they plant something better. Next slide. So, and this last one, so technically I should cover this because it's a section of plant pathology. There are things called phytoplasmas that are not of the fungi, bacteria, and viruses. This is peach yellows. It's a phytoplasma. It's kind of, if you want to know what a phytoplasma is, you can think of it as a highly reduced bacterium or sort of a virus on steroids that actually might be kind of living. They are few and far between. They generally don't cause much in the way of economic damage, but for instance, something like peach yellows, if you get it in your peach trees, there is no treatment, and you have to cut down the peach trees before it spreads to all the other peach trees in your orchard. So it can be a big problem in rare instances. That said, we don't run across them much in the landscape. So we're not going to spend a lot of time talking about them. Next slide. Okay, so this is what we do run across. And if anybody wants to type into the chat what they think this is, please do. Anybody feeling adventurous? Does anybody know the plant? Are you guys limited?
A rhododendron of some kind?
Yeah, very good. So somebody said it's a rhododendron, and you got that right. Okay. Rust is a bet, and the reason you might think it's rust, partly because rust is named after the rust red color that it does produce on plants. So that's not a bad one. Somebody else is guessing frost damage, and that's not a bad one either. But Linda gets it right. Extra points to Linda, because this is sunburn, sometimes called sun scald. And the way you can tell this is if you look fairly carefully. Now, I don't have a pointer on this one, so you guys are going to just have to bear with me. But in the middle of the right-hand side of that image, you can see a leaf that has a burn on the edge on one part and a burn in the middle on another part. You guys see that? Susan's got the cursor on it right now. Why do you think that's happening? It's because the sun is hitting it at shaded. Okay, so yeah, Stephanie got it right. It's because of the way the sun's hitting it, or the flip side of that coin is the way the shade is. So the sun's hitting at a fairly low angle on this particular one and hits along that edge. So if you can imagine the curvature on that leaf, there are certain parts of the leaf that are hitting the sun's hitting straight on, and there are other parts of the leaf where it's sort of rolling away from the sun, so it's in the shade slightly, and that's enough to cause it to not burn. And that's a clue to us when we're looking at this, that this is really more of a sun issue. And so the reason we're talking about this is it's an abiotic plant decline. Yeah, you might end up getting bacteria or fungi or something growing on this a little bit later, but the proximal cause of the problem is something that was not a bacteria or a fungus. But if we're going to really be honest, well, actually, I think we'll get into what is abiotic. Yeah, so for the chat questions, this is going to be a long talk. So let's take the questions as we get them. That would be lovely. Do we have one, or?
Stephen, do you want to go back and hear some of the earlier questions? We don't have
absolutely. A lot. If they're, especially if they're points of clarification, I really want to hear them, because otherwise I don't know what I'm skipping over, and so the class keeps me honest, you know?
Okay. Well, Jeff asked earlier, so mycelia is the mass of hyphae?
Yes, absolutely. So Jeff got it exactly right. Mycelia is a mass of hyphae, and the people will be start using these terms around, and they can be really confusing. So thanks for that question, Jeff. You got it.
And Olivia asks, would plant material afflicted by armorillia be killed in a hot compost system?
Probably. Armillaria is one of the tougher customers out there. Fusarium's even tougher than armillaria. So there are, for instance, no chemistries that will help you deal with armillaria. In a hot compost system, if it's done right, yes, armillaria should be killed. The problem is if it's done right. So yeah, a commercial compost system generally will do it right. The problem is when you build a pile, the toes of the pile, those outermost edges typically don't get all the way to 131 degrees Fahrenheit. So unless somebody's very thorough about turning and making sure all parts of the material that's in a compost system get to 131 degrees, and that means you got to turn it and get it to heat up again and then turn it and get it. And when I say turn, I mean you're turning those toes of the pile into the middle so that every part of the pile is eventually exposed to 131 degrees Fahrenheit or higher for at least a few hours, because that's how long it takes to kill at 131. And different composting systems, and we can get that, it's outside of what we're going to talk about today. Different composting systems have different kill times because of the way they work. But suffice it to say that a properly run hot composting system should kill armillaria. It will not necessarily kill all strains of Fusarium.
Okay, David Elliott asks, in the case of this sunburn on the rhododendron, will it weaken the mature plant in a meaningful way or just unsightly?
That's, we're picking points on a curve. If the plant gets seriously sunburned and it loses an awful lot of leaves, it's just a question of you know, how bad is the damage? And so the question is yes, the answer is yes. It will either seriously damage it or it's just unsightly, depending upon a matter of degree.
Okay, Stephen, that's the questions for now.
Okay, good questions. Thank you. Kay, interrupt me at any time, because I would much rather have this be I know it's absurd to pretend this is really a dialogue, but I much prefer a dialogue than some talking head just going on and on and on. You feel like you're watching a TV show. So, so please interrupt me and we'll keep going. Okay, so next slide. Okay. Okay, great. So this is abiotic, and it's because, and we'll get into this a little bit more, it's herbicide damage. And that should give you a clue of where we're really going with abiotic in the long run is it's usually not so much a problem with a plant. And we'll just leave it at that for right now. Next slide. The other things that we study in plant pathology include parasitic plants. Now this is really kind of cheating because it's not plant pathology in the classic sense, because it's not a microorganism. I mean, look, you're looking at the plant right there. So this is a problem. The thing is that some of the parasitic plants live internally. So some of these mistletoes you can't see from the outside. They are internal to the plant and they start behaving kind of like a microorganism. So nobody else wants to study them. So they're what plant pathologists end up studying. So we're the dumping ground for all the stuff that nobody else wants to deal with. And the next slide. And nematodes. Again, this is something that often cannot be seen outside the plant. And even though it's not technically a microorganism, because you can see nematodes if you pull them out of the plant, once they're inside the roots, you really can't see them, and nobody else wants to study them. Now, nematology is its own discipline. And so UC does have a few nematologists, but they are so few, there's only like 3 or 4 in all of UC that do plant nematodes, that it can be challenging getting the answers you need out of them sometimes. And I'm not trying to talk them down. They're just busy and they've got a lot of things going on. So, but plant pathologists end up doing a lot of work on nematodes because main reason being is when you've got a declining plant, they generally don't take it to a nematologist. They take it to somebody who can diagnose plant diseases. And most of the time that's fungi. So you get this declining plant, you rule out all the fungi and you start saying, I think maybe this is a nematode. And then, so anyway, plant pathologists play in nematology a fair amount. Enough said. We're not going to spend a lot more time on nematodes today, unfortunately, because it is a pretty cool topic. Next. Okay, so I just covered a whole lot of stuff that, plant pathologists study that aren't really microorganisms necessarily. That includes, you know, climate, sunburn, nematodes, and all kinds of other stuff. So what don't plant pathologists study is almost easier to answer. So vertebrates, we don't work with gophers. So that's really the, you know, when we're talking about vertebrates, it's usually wildlife biologists or veterinarians. We have a couple vertebrate control advisors within UC. So if you've got questions that are really specific and detailed on vertebrate control, whether it's blackbirds or gophers or deer, we can talk to somebody at UC who knows something about them. And then, you know, the whole domain of invertebrates. Next slide. And they're both animals. So basically, what don't plant pathologists study? Well, animals that you can see. And this is, you know, vertebrates and invertebrates. And this is all the insects primarily, although mites and things also get put into entomology. So, yeah, enough said. Yeah, next slide. Okay, I could have sworn we did, yeah, we did this for IPM. We're gonna skip through some of these next couple of slides because you guys have already seen this. We've already talked about cottony cushion scale and the Vidalia beetle and how it came in, and this is not, and that's part of IPM as well. Next slide. Next slide. Okay, and we already covered this in the last lecture, so next slide. Wait, is this my IPM talk? Did I send you guys the wrong thing? Geez, keep going. Yeah, this looks like the IPM talk and not the plant pathology talk. I am so sorry. So I've got a problem here, and I'm gonna have to that explains that. I thought this looked an awful lot like the IPM talk and the plant pathology talk, but I need to fix this. If you guys will give me 5 minutes, let's take a break. I'm gonna put another slide up, and I'll try and share it from my screen. And what I'm gonna ask, and we'll see if we can't run it that way. If we can't, then I will send it to Kay and Susan, and they can fix it from their end. And my apologies, this was entirely my fault. Kay and Susan have been doing a wonderful job, and this has not, they're just the bystanders in the train wreck I created.
But if you can talk about the disease triangle, that would be helpful.
I'm sorry?
If you can talk about the disease triangle when you do your talk, that would be helpful for the class.
Okay, can do.
Okay,
I just gotta go dig up my, I got my flash drive right here. It's not my flash drive, it's my, the hard drive that has all the data on it. And I will plug that in and we'll get that.
Okay, why don't we take a break until 10 o'clock?
Sounds good.
Okay, thank you.
And here we go. Where is autoplay? View files, working documents, master gardener, presentations, IPM and pathology. Okay, Susan, or Kay.
Yeah. Yes.
Is it possible for you guys to give me the screen?
Yes.
'Cause
Anna will arrange to give you the
I just want to try it and test it.
Anna's the one who has to give you the screen.
Yes, I'm giving him the co-host option.
Thank you.
There you go.
Good, it looks like I've got it now. So if I bring up my things that kill plants, Plant Pathology 101, and I go here, and then how do I get my escape? Well, how do I do that?
Okay, let me.
I got to go into Zoom somehow and go share screen,
Anna's coming to rescue you.
right? I got it. I think I got it here. And no, it's okay. Come on in. I'm not gonna, now I just got to find the right control. Slideshow. Come on. Slideshow from current slide. Now, are you guys seeing the things that kill plants 101, plant pathology?
Yes.
Okay, good. So we're ready to go. We've already, as you can see, there's a reason that it all looked the same because there's a lot of the similar slides. But when we got to,
So Stephen, they're still on break for another 10 minutes.
yeah, no, I'm worried that what's happened is I saved it under the wrong name.
Okay. Please mute yourselves and we're back to plant pathology.
Okay. So the disease triangle. This is a moderately useful way of thinking about plant disease. It has its limits. Any little model does, but this works okay. And we'll talk about where the instances where it doesn't and why. Okay, so in order for us to have a plant disease, it's a little bit like the fire triangle. You need three things, right? You need a plant, which in this one is called the host, and this host can vary in its susceptibility. And you need the pathogen, obviously, and the pathogen can vary in its virulence. But you can have the host and the pathogen in the same area together, and you don't get disease all the time. That happens all the time with even really well-known killers like armillaria. You know, armillarias on, if you go and take samples of dirt from, you know, schoolyards, freeway sides, farms, wilderness, people's lawns, people's gardens, places like, if you take just 100 samples out of random places, you'll get armillaria out of like 40 of them. I mean, it's everywhere. But it's not killing plants on 40% of the landscape. It's only killing plants in a few isolated instances. And that's because the last one is the environment. So the conditions have to be right to cause disease, and the host has to be stressed for armillaria to really get started. So, you know, the presence of the pathogen, it's just waiting there half the time for both the host susceptibility to be high and the environmental conditions to be right. And when both of those things occur, then you get disease. So, so we'll talk about the one or two conditions where you get disease in the absence. And so like pretty much abiotic stuff is a condition where you don't necessarily have a pathogen. You have a host and the environmental conditions are so bad, they cause disease. So we'll talk about that as its own sort of separate issue here in a minute, because I still think there's a disease triangle, even though we're missing one element, and the pathogen part gets a little tricky. Okay, so let's talk about, we've given you all kinds of historical examples and, you know, and where does plant pathology come from, and you know, what are we really talking about? We're mostly talking about abiotic disease and maybe a lot about fungi and maybe a few bacteria and viruses and maybe in rare instances some other stuff. So in order for you to do diagnosis, what do you need to know? Well, the first thing is identify and research the plant in question. So, are the symptoms normal? Here we have an image. Anybody, does this look right? What is this plant?
You. It looks like rosemary, maybe?
It does look like rosemary, but it's not. It's a tree, I'll promise you that. It's big. It looks kind of like a pine tree. Should pine trees look like this?
Yeah, but not healthy ones.
Depends on if it's a deciduous conifer.
So what kind of deciduous conifers are there?
I think there's a cedar variety, but I can't remember offhand.
Larch.
From the,
No, but you're on the right path. Really well done. So this is a larch. It's a deciduous conifer. And if you work the desk long enough, you will find some homeowner, because we have enough of them in Marin, where some poor guy buys a house and then, and he typically buys it in the spring or the summer. And then, 3 months in, the plant turns orange on him and drops all of its things. And he's like, I just bought the house and the plant's dying. What do I do? And it's like, you got a larch. It's normal. So, we don't usually think of conifers as being deciduous, but there are a few. Dawn redwoods are another example. So those questions, thank you to those of you who gave it a shot. That's how we're trying to get through things. So this is absolutely, in this particular case, is absolutely normal. This is a larch, and it's going to drop all its needles, and it'll be fine next spring. So this would be sort of a symptom if it was a pathogen. It's a plant response. So that's what we're going to talk about two things, and you guys are going to vote here for what you guys think it is, a symptom or a sign. A sign is a visible pathogen structure or indications of activity. So this can get a little confusing. And so we're going to start off with some fairly easy ones, but there are going to be some tricky ones in here on whether it's a symptom or a sign. So feel free to play along if you want. If you just want to watch, that's okay too. Next slide, please.
Stephen, you're doing the slides.
Oh, I knew that. Sorry. Oh, now this will make things go much better. Faster, I mean. Because now there. Okay, so anybody know what we're looking at?
Peach tree.
Really close. Yeah, it's an almond, right? What are we seeing here? Are these symptoms or signs?
signs of fire blight.
Okay, so everybody agree it's a sign of fire blight?
Symptom.
I agree it's a sign.
I agree it's a sign
So why is it a sign, guys?
because it's visible.
Okay, visible
The
pathogen
plant-
structures. So it is visible, but plants' responses to a pathogen are visible too. I'm going to make the argument that this is a symptom. This is what you're looking at is just the plant response. The pathogen got in there, the plant's dying. You're just looking at a plant response to the pathogen. That's okay. If we get it wrong a couple times, you'll get the hang of it. So what are we looking at here?
That is signs,
Signs. Why is it
Yeah.
a sign?
because you can see the pathogen structures here clearly.
Yeah, right. And so, so this is turkey tail or Trametes. Not that you care, but turkey tail on apple tree. It's a fungal disease. It's a wood rotter mostly, but when it gets big and mean enough, and this is when it's big and mean enough, it can get aggressive enough to start killing living tissue. So this got in probably through a pruning wound or something like that, and the tree was not in good health, and now it's taking the tree over. So this is a sign. Now, you know, occasionally people bring up the question, well, wait a minute, you know, what's the difference between a sign and just looking at, you know, it's the term sign comes from, you know, when people did animal tracking. So you'd see a footprint in the ground, or you'd see poop on the ground, and you'd say, oh, that's a sign of a bear. Because look, see, it's a bear hoof, paw print, or it's bear poop. So you'd say that's a sign of a bear. At some point, we're looking at the bear here. The Trametes, that you're seeing, that is, you know, it's like, at some point, the whole idea of sign starts breaking down a little bit, because, you know, you're just like, oh, look, there's a footprint. Oh, look, there's bear poop. Oh, look, here's a claw. And look, here's a whole set of teeth. You know, this is a sign of a bear. At that point, you should be running, because that's a whole set of teeth. You're looking at the bear, and he's going to eat you. So the reason I'm bringing this up is the difference between a sign and just looking at the pathogen, it's blurry. And in this case, I think we're pretty much looking at, now, granted, we're not looking at the whole pathogen, we're just looking at the reproductive structures, but that's pretty much it. Okay, so symptom or sign, anybody want to take a wild stab as to what we're looking at?
Symptom.
Symptom.
Looks like a sign of some vertebrate damage or something.
Okay,
Sorry, invertebrate, not vertebrate.
sure. And I'm not going to disagree with you. That could be a possible, that's a possible sign. If it was invertebrate damage, then this would be a sign of invertebrate damage. I agree. But that distortion on the leaves is not going to be caused by well, that's not true. It could be caused by green peach aphid. In this particular case, if I remember correctly on this one, this is sublethal herbicide damage. So this would be a symptom because it's simply a plant response. So this is where things are getting tricky, but it was a good shot. You know, if this had been green peach aphid or something that ate, then it could be a sign. What about this one?
Wow.
So first of all, do we even know what we're looking at? Nope. Okay, so this is an oak tree that is snapped, and we're looking at the interior of an oak tree, and we've got these black things going through the interior of the oak tree. Those are tunnels. Are these symptoms or signs?
Symptom.
Sign.
Okay, sign. So it's not a plant response. This is something the insect did. It created those tunnels, right? And then, because this is an ambrosia beetle, those tunnels are black because a fungus has grown in those tunnels because the beetle isn't actually eating the tree. The beetle's just creating tunnels, and then it's farming by putting fungal spores in it, and the fungus is eating the tree. The fungus is then growing, and its spores are lining the tunnels, and the insect is eating the spores. So you're looking at signs of the fungus, and you're looking at signs of the insect. Okay. This one's challenging. And so if you don't know what it is, if you do know what it is, please holler. It's not impossible. We're looking at the bottom of a leaf. Anybody think they know what these might be? They're galls caused by an insect that lays its eggs inside the leaf tissue, and the enzymes released by the egg cause the leaf tissue to grow scar tissue that's bright red and envelops the egg, and then the insect eats the scar tissue. These are called galls, plant galls, and this is on the bottom of an oak leaf. And I'm not sure whether they're symptoms or signs either. Because, you know, you could make a good case that, you know, these are structures sort of almost caused by the insect. At the same time, they're definitely a plant response to the egg being laid in the and the enzymes. So that could be either way. Does that make sense? Okay. This is a surface of an orange. That's what you're looking at. Are these symptoms or signs?
Sign,
Okay, what's the sign?
the fruiting black bodies of what appear
Good.
to be a fungus.
Are there symptoms here too?
Possibly.
What would the symptoms be?
The rotting of the fruit.
Very good. Who's saying that, Emery?
That's me, Jeff.
Jeff. Ah, got it, Jeff. Good, extra points for you, Jeff. You got both of them right. Well done. So the signs are the fruiting bodies. So these are the fungus is producing these little black flasks, if you will, that are embedded in the fruit, and they will exude spores if it gets wet. So those are the fungus, the tips of the fungus actually just sort of peeking out from the surface of the fruit. But the fruit is the fruit should be that bright yellow orange color, and as it fades to orange to brown, that's a plant response to being digested by the fungus. So you're looking at both symptoms and signs in this one. How about this one?
Symptom,
Why symptom? Everybody agree?
lots of excessive new growth from the plant in response to something.
Yeah, exactly. So these are symptoms. It's pretty straightforward. There is no sign. We have no idea whether this is an herbicide damage or it's feeding by areophyid mites or any number of different things that could cause this kind of thing. But we can see a plant response that's not normal, and it's not necessarily healthy. So that is a symptom, absolutely. Okay, so this one is going to require a little bit of explanation. What we're looking at here is a close-up of a eucalyptus leaf that was growing in Sonoma Square. So most of the leaf looks pretty healthy, but it's got all this blistering. And these are getting challenging now. So don't feel bad if, anybody want to take a stab at whether this is a symptom or a sign? And if anybody feels like they're particularly brave and wants to say this is what they think is going on, let's hear that too. Okay, well, the problem here is abiotic. There is no pathogen or insect involved. This is simply caused by, have you guys ever looked at the grass in Sonoma Square? No? It's pretty lush, right? So Sonoma Square gets a lot of people in it, and it's got a lot of shade, and it's beautiful, and the grass is lush, and you can picnic on it, and it's very well irrigated. This is a eucalyptus tree that's adapted to dry environments. It's in Sonoma Square, and the water pressure inside the leaves is getting so freaking high, it's causing cells to burst. This is called edema. It's abiotic. There is no pathogen causing this. So it's got to be a symptom because there's nothing to create a sign. Does that make sense? Okay, well, we're just trying to get through the ideas. Okay, so this is the bottom of a rhododendron leaf. Is this a symptom or a sign? And I'll give you a clue. Should rhododendron leaves be hairy underneath?
Sign.
Sign. Somebody said it's a sign. Let's hear from who said it's a sign and why.
You can see, this is Emery, you can see along the stem, I think you can see some hairs of all kinds of a little lighter color than the normal leaf.
Right. So you got it. All that hairy looking stuff is powdery mildew. And sometimes on rhodies, powdery mildew will only show up where you get, when you got to look at it from an angle under the scope. So if I had taken the same shot face on the leaf, the fungal mat would be so sparse you could barely see it. In fact, it probably wouldn't have shown up in the photograph hardly at all. one of the evidence for that is there's a little bit of it on the green vein in the middle, and it's hardly showing up. I mean, but so the only way you really see it is looking through it at an angle. So this is powdery mildew. So this is a sign. You're not looking at a plant response to anything because the plant still looks pretty decent. It's green, but the fungus is there. Okay, so we went through the symptoms and signs. I hope you guys got a feel for that a little bit. A symptom is a plant response. A sign is something other than the plant response, a direct response. So you're going to see a lot of abiotic problems, and you're going to see quite a few fungal problems. So that's what to expect if you're working the desk or something along those lines. And you will see perhaps a few bacterial problems. Notably, it's going to be in the winter that you'll see the bacterial problems show up because they're going to be, or maybe the spring, depending upon how the weather's been. It'll be fire blight and Pseudomonas, frost damage, those kinds of things. And you'll also get a few viruses that'll show up from time to time. And rarely will we see phytoplasmas and plants. And at least rarely will we be able to identify nematodes. Now, I am told by a lot of people who are nematologists that nematodes are much more prevalent than people think. And they cause a bunch of things, but they're often misdiagnosed. And I'm going to be one of the people who misdiagnoses them because they're pretty hard to do. So we may actually see more nematodes problems than we realize, is all I'm trying to say. Okay, so that's what to expect. Let's, we can do a little bit of the symptoms and signs. And here, the next thing is, sometimes this stuff, it gets a little hard to assign agency to. So anybody know what we're looking at here? Anybody think they know what we're looking at? What do you guys see?
Crown rot of some kind?
Yeah, crown rot. So this is a walnut, and walnuts have bright, healthy bark on a walnut. It's usually a pretty bright yellow. So that's what we're looking at, is bright yellow bark on a walnut, except the bottom's not bright yellow, is it? So this is, my best guess would be it's a Phytophthora. I believe that's actually what it is. And we're looking at Phytophthora infection of the root crown of a walnut, and it's working its way up the stem, although it won't have long to go because at this point the tree's pretty much dead. There's nothing left alive on the roots, so the rest of the tree is just going to go. So this would be a symptom or a sign?
Both.
Okay. So are you seeing the actual pathogen here? Phytophthora are microscopic.
No, I guess not.
No, it's a good, no, it wasn't a bad shot. So mostly what we're seeing here is a plant response, and the plant response is it's dying as a result of the infection, right? Good. Okay, so that's a fungal problem. This is a bacterial problem. Anybody know what we're looking at? Want to take a stab? It was mentioned earlier by one of you guys. This is fire blight. How do we know?
Looks like it's been burnt.
Yeah, it looks like it's been burnt. And the leaves are just emerging. If it's fire blight, it only attacks apples and pears, right? And well, and a few relatives like Cotoneaster and Pyracantha and Toyon. So this attacks only pome fruits. So pome fruits, apples and pears, they bloom first and then the leaves come out, right? So this fire blight comes in. I'm not expecting you guys to know this. So I'm just sort of using it as this is how we're thinking about how we're approaching this. The leaves are just showing up. That means it's already finished its bloom. And look, the bloom, the little buds that carried the flowers, it's all withered, black, and dead. And now the bacteria has gotten into the trunk, and now it's moving its way up and down the trunk. And now you've got bleeding coming from the trunk where the tree is trying to fight the infection off. and it's just starting to bloom. So you know what time of year this is. This would have been last month when your apple trees were just putting on their first round of new leaves. So this is fire blight. And this is tobacco mosaic virus on tomato. Yay! So we will see a few viruses. It's not all that common, but we will get it. Particularly, you know, tomatoes they've got a lot of viruses. So we'll see it on tomatoes and roses and a few other things that people grow a lot. Okay, so abiotic diseases. The reason I wanted to bring back to the disease triangle is that abiotic isn't really abiotic. It's just that when people bring stuff into the desk and say, what's the problem? You get to tell them it's an abiotic issue because that's a very nice way of saying the problem is you. Abiotic is a way of saying somebody screwed this up. Because abiotic problems don't often occur in nature. They're usually the wrong plant or the wrong place or both. Now, that said, you're right. I can hear a couple of the voices already going off in the backs of your head saying, you know, we live in California, buddy. There's such thing as drought and trees die from it. Yep, you're right. You're right about that. So we do occasionally have plants that die from abiotic, or at least develop abiotic symptoms, and they don't always die from them. But a lot of times what we're looking at is people overwatering or underwatering, or in the case of this poor rose, this is 2,4-D damage from accidental herbicide exposure when it drifted through. So abiotic is basically a way of telling people that they a very nice way of saying you screwed up, usually. There are all symptoms. There are no signs. There are rare exceptions to the no signs thing. Sometimes you get the evidence left on site by the people who did it. Leaf symptoms are almost always seen first, as we see here. This was herbicide damage, yeah. So abiotic diseases. This is an oak tree. What happened here? Any guys? There's a few. There's actually signs of what went on here. Although it takes a little bit to put it together. Anybody want to take a stab at this? What's going wrong? What are the symptoms that the plant's exhibiting?
Well, it's planted close to the lawn, so it might be overly irrigated.
Yeah, Terry, good. That's well done. So a little bit of history here. That lawn wasn't always pulled that far away. That plant was put in that lawn, or rather that lawn was put around that plant, and the plant started to die, and they sat down and said, what's wrong? Well, so as Terry would probably be able to tell us, you don't want a lawn around the base of an oak tree. It causes all kinds of problems. The oak doesn't like summer water. It doesn't like warm, wet conditions around its soil. So the oak started to decline because it developed root disease. So then the people that put the lawn in asked themselves, whoa, we got a problem. Our oak is dying. What do we do? And they came out and somebody said, oh, you got a root disease because you've over irrigated this. So then they ripped all the lawn out for a long way around the tree. So let me ask you this. If a tree develops a root disease, how much access to water does it have?
Substantially less.
Substantially less. Why? Because the roots aren't working, right? So now the roots aren't working. Now you're going to try and fix the problem by ripping out all the lawn and making sure it doesn't get any water, right? What's the problem with that?
Suddenly not getting enough water.
Yeah, that plant was already not getting enough water because its root was diseased, and now you're making sure it doesn't get any water. This tree recovered. I mean, oaks have a lot of reserve energy. So, you know, it's okay. I mean, it ended up working out. But what I'm trying to say is in abiotic diseases, there's a little bit of a story here, and part of it's told by that fresh new lawn all around it, and then the very lack of fresh new lawn right around the base of the tree, and not even mulch, you know. Anyway, so those are, I kind of consider those signs, you know, thinking about how we do this. Okay, so this is a poplar. It's abiotic. Anybody want to tell me what they think it is, maybe? This is herbicide damage on poplar, 2,4-D again. Okay, so how do we figure this stuff out? So for the most part, we're going to go to something called UCIPM. And for herbicides, it's really hard to figure this stuff out. But when you go to our IPM website, there's this, can you see the red circle around the herbicide symptoms photo repository? So there you can, if you've got questions about maybe this is an herbicide because you got these really distinctive symptoms, and herbicides and viruses can be challenging because they both produce these foliar symptoms primarily, and they're very distinctive. You can go through, and a lot of these things you can look up because they've got tons of examples of where people have misapplied this, that, or the other thing, and some where they've intentionally applied the stuff so that you can see what the symptoms look like and say, okay, so this is what glyphosate looks like on rose. This is what glyphosate looks like on Douglas fir, you know, or this is what glyphosate looks like, accidental glyphosate looks like on strawberries, all kinds of things. So you can look these things up. I'm not going to tell you that everything's always there. And the problem is sometimes when you're trying to figure out, you know, hey, is this herbicide on petunias? Particularly, is it, you know, any one of a number of, you know, propiconazole on petunias. Good luck on finding propiconazole on petunias. It might be there, but you may have to extrapolate from something else. You know, the tools are there, but they're still necessarily a little bit limited. Okay, but not all abiotic diseases are caused by herbicide. In fact, even though those are great photos to show, the vast majority of them are not caused by herbicide. What is the biggest problem with abiotic diseases? Anybody want to take a guess? What is it? What are you most likely to see?
Over or underwatering.
Very good. Excellent. Watering. It's almost always a watering problem. And that's, again, it's abiotic, and it's usually that somebody messed up. So symptom uniformity is characteristic. Now, I've got uniformity in quotes because when I say uniformity, it doesn't mean that every leaf on the plant is showing the same symptoms. But maybe all the older leaves are showing all the same symptoms, or maybe all the newer leaves are showing the same symptoms, or maybe all the leaves on the southwest side of the plant are showing the same symptoms, and all are going to tell you something something slightly different is going on. Does that make sense? So look for patterns in the way that the symptoms exhibit themselves. So, for instance, when we get marginal leaf burn, it could be a number of different things, including drought and salt injury, or potentially herbicide and others. Marginal leaf burn is usually caused by either drought or salt injury. And of course, those two are related too, because when you look at water in soil, as the plant takes up all the fresh water it can get out of the soil becomes slightly saltier and saltier and saltier. And at some point, the plant, rather than die of thirst, is going to start bringing in some of that, a little bit of salts into into the plant too. That's almost inevitable. So plants will take up a little bit of salt. Once the salt's in the plant, the water is all evaporated through the leaves. What happens to the salt in that water? Where does it build up the first? Where is the evaporation the greatest on a leaf?
Margins.
Yeah, on the margins. That's where you have the greatest surface area to volume. And once the salt moves all the way through the leaf and it gets out to the end there, that's where the salt all builds up, because there's no water flowing back out of the leaf back to the rest of the plant, so it doesn't get diluted going that way. So you get this salt burn showing up on the tips and margins all the time. So drought and salt injury go hand in hand. Often you can't tell one from the other. Now, sometimes you can. Sometimes the plant has plenty of water, but somebody loads it up with so much fertilizer that you develop marginal leaf burn, and it's not really about drought. And sometimes there isn't that much salt in the soil, and the plant simply runs out of water, and it'll burn from the tip in anyway, just because that's where the evaporation's the highest, and it's not really about salt. Anyway, so what we're looking when we're trying to think of these things, I'm just trying to help you think about how we might approach a marginal leaf burn question and say, what are we trying to figure out here? What are the likely causes? So symptom uniformity is characteristic. There are some exceptions. Some exposure to sun and frost may not seem so logical, especially when you get sunburn caused by the removal of shade. So this will be a plant that's grown its whole life in shade, and then somebody cuts down the shade of a tree, and then that tree dies and somebody cuts it down, and all of a sudden it gets blasted with a whole lot more sunlight than it's used to. But you look it up and you're like, well, you know, it should be able to handle full sun. It says full sun in the Sunset Western Garden book, and you know, it's not like it's all just coming from one spot. The plant's just fried. I don't, you know, and that's just going to be, that's only going to come out when you talk to the person and get some of the history. And frost can be similar, you know, depending upon the details. And then, you know, you get things like irrigation and construction. These are technically, we call it abiotic. Oh, I'm sorry, and I'm getting back. I've skipped a few things. So here we have, this is salt burn in avocado, but we're going back to this. So that's drought and that's salt burn. They look remarkably similar. Again, 2,4-D damage in poplar. This is nitrogen deficiency in avocado again. And this is all backwards. What's wrong with me saying it's nitrogen deficiency? Why does that not fit the model that we teach? What do we usually see when we see a nitrogen deficiency? What is the greenest and what's the what stays green and what turns yellow when it's nitrogen deficient? When a plant's nitrogen deficient?
The new growth is usually green, and the old growth usually starts to yellow.
That's exactly right, Jeff. So what Jeff said, I think everybody probably heard, was that the new growth should be dark green and the old growth should be yellowing. And that's the classic sign of nitrogen deficiency. But here I am saying this is nitrogen deficiency in avocado, and the new growth is pale green and the old growth is deep green. So that's all messed up. It's backwards. How can this possibly be nitrogen deficiency? Anybody want to take a wild stab at that? So let's think about, the citrus shows this same color pattern in the spring in Marin and Sonoma. And both avocado and citrus are subtropicals. So they usually move nitrogen just like every other plant. But the problem is they're used to warmer temperatures when they're putting out their new growth flush. And with when it stays cold when they're putting the new growth flush, the plant has a hard time moving nitrogen. So the plant shows up being chlorotic. And the new growth is nitrogen starved. That's why it's yellow. But the problem isn't necessarily even the lack of nitrogen. It's just that it's so cold right now for a subtropical plant that it's just having a hard time moving it. Is that making some kind of sense? I'm bringing up a lot of exceptions, but I think exceptions help teach us the rules. So I'm hoping that's helping. I'm not expecting you guys to memorize all this. I'm just bringing this up as ways of thinking about stuff and bringing up some of the exceptions that you're going to run across. So avocado and citrus are going to show up having yellow new growth this time of year pretty much in all the time in Northern California. Now that may change as global warming keeps going. We can look forward to some of the few benefits of global warming, and one is our citrus will do better here. But whatever. Okay, so other abiotic diseases and symptom uniformity. This was actually caused at an experiment at UC Davis. They took a liquid amber tree, so those are all liquid amber leaves that you're looking at, and they dumped a bunch of lime on the thing to raise the pH up to about 8, 8.5. So the old leaves were at the bottom, and in the middle are the leaves that were emerging as this happened, and then the top is the newest leaves that emerged at pH 8.5. Anybody feel like they know what might be going on here?
Iron deficiency.
Very good, iron deficiency. So those green veins. Now, granted, magnesium deficiency and several other. I think molybdenum is also has green veins and yellow intervenal areas. So, but you're right, this is iron deficiency. It could be one of any, but the point is that you get a nutrient deficiency caused by a pH shift in the soil. So good. So we've already seen this once before, but this time it looks a little different. So what's this?
Sun damage
Very good, sun damage. And you know that because?
of the, where the, where you see the brown spots.
Right, where the shade is, where the sun hits.
Right.
And it's pretty much right there. So a little bit of sunburn. The reason I put this in here is that woody leaves, tough, thick, leathery woody leaves tend to show sunburn that burns like this. Softer, juicier leaves tend to show sunburn that looks like this. I don't even remember what this is. Maybe one of you guys knows what this plant is. Something with a nice red flower and soft leaves that burn. And you'll notice that this in the old, this case here, it went right over those leaf veins. And here it's only burning between the leaf veins. Or, you know, it's also burning the leaf veins, but only at the very end. Okay, some more abiotics. This one is taken, this is a pear leaf that's just emerged. That tells you something about the time of year. Anybody want to guess what the problem is?
Is it a fungus?
Leaf curl.
It's not unreasonable. We do have fungi that cause symptoms that look kind of like this. Notably, some of the powdery mildews can cause a little bit of stippling, not usually this severe. But no, this case is not a fungus. But you'd think that because of winter rains, maybe that would be the case. Anybody else?
Leaf curl.
Leaf curl's a fungus.
Oh.
This is sublethal frost damage. So what happens here is that the ice crystals form, but it's not bad enough to kill the leaf. It causes, as the ice crystals form, it causes cells to burst. As the cells burst, the plant forms scar tissue. So it makes it, and as you know, scar tissue isn't as flexible and stretchy as normal tissue is. So as the leaf is still expanding, the scar tissue causes distortion in the leaf as it's trying to grow out. So this is sublethal frost. This is also frost damage, a little bit more typical. How do you tell this is frost damage?
It's on the exterior leaves, not down in the center of the plant.
Yeah, all the leaves down in the center are fine. It's happening in winter. You know that because the oranges are ripe on the tree. And you've got the stuff that's sticking up above where it's not shaded. The shade works, it's not sheltered from the cold air by other leaves. The company of other leaves helps keep things a little warmer inside the canopy. The stuff that sticks out is the stuff that gets burned, frost burned typically. So good. Again, freshly planted avocado out in the middle of things. There's no shelter for it. You get a sublethal frost and it burned all these other things. The avocado's still struggling back though. Okay, so, and then we get into some of these other questions. So this is probably fungal. It's probably a Phytophthora, although, yeah, I can hear if one or two of you guys saying Phytophthora is not a true fungus, but we're going to call it a fungus because it doesn't fit into the other categories so easily. But sometimes this can be caused by overwatering, right? So this was a plant that I believe was intentionally overwatered to sort of illustrate this point. And how do you figure out whether this was abiotic or biotic if you've got a plant that is overwatered and then develops a root disease as a consequence? You know, because we can point at it and say, yeah, that's a Phytophthora infection, but it would probably have never gotten that if somebody hadn't overwatered in the first place, if that makes some kind of sense. And then we get a little bit more extreme cases, abiotic, and, you know, I mean, I don't know what anybody's expecting this tree to actually do when they're done at the end of this construction. But it's not going to be good. They'll be lucky if it stays standing. Anyway, yeah. So abiotic, the problem is us. So what's going on here? Anybody want to take a, here's a hint. Look at that nice new wall with the newly planted shrubs right around the base.
Mm-hmm.
And you've got two redwoods that are usually pretty tough.
Roots are
Roots.
Yeah, roots, exactly. So in order to put a nice big wall like that up, you need a foundation for the wall, and that means you're digging down. And what had to happen when they dug down right next to those trees? They severed probably at least almost half of the roots of those trees. That wall was probably put in the summer, and voila, we have no more water conduction, or at least not enough water conduction. So to treat abiotic diseases, what are we doing? We're altering to favorable environmental conditions if we can. So how would we go about fixing this? Or how would we go about fixing this? I'm not positive you can, but you know, the thing to do would be to backfill and provide enough water so that the plant doesn't starve using its remaining root systems, and then pray you don't get a Phytophthora like we got here. I mean, this plant didn't even have its root systems cut. If you overwater it, it develops a Phytophthora. Here, you're going to have to put a ton of water down just to get the plants to have a chance at recovery, but there's a good chance you'll develop a Phytophthora anyway. So, you know, the bottom line is don't do bad things to plants and you won't have to fix this. Okay, so let's talk about Basidiomycetes really quickly. Do we need another break really quick? I'm just checking in with Kay and Susan.
I don't know, Stephen. It's pretty interesting. We can ask the class. Break, no break.
How you guys feeling? You guys want another 5 minutes, or should we keep going?
Keep going.
Keep going. Okay. So, so fungi. We're going to start throwing some more terms at you. Fungi are grouped into a number of different groups, and one of the more common one is Basidiomycetes. So you'll see in the sort of lower center of the this image, you'll see this thing that looks like a bear paw. It's got 4 little spores sticking out of it, and that is a basidium. And those are basidiospores, or the little toes on the end of the bear paw. So that's where it gets its name from. Some of the basidia don't look like bear paws. A lot of the basidia look like trees. And those are what have the spores, and those spores are, these basidia line the gills of the mushroom, and then the spores line the basidia. So the spores are just absolutely tiny. But this is what gives this group a name. And most of the Basidiomycetes are mushrooms. Now, you don't have to remember this. This is not going to show up on a test or anything like that. All I'm saying is one group of fungi are the Basidia. We're going to cover the big groups of fungi and say, and this is the classic mushroom we all think about. So Armillaria is a Basidiomycete. It's a plant killer. Most basidiomycetes aren't plant killers. A lot of them are actually plant helpers. You know, things like mycorrhizae and that kind of thing. So here's Ganoderma. This is a basidiomycete. It is a plant pathogen, but that's what I do. I tend to take pictures of those. This grows on the sides, so you can see it doesn't form a mushroom. It forms what's called a conch, but it's still a basidiomycete because it's still forming these structures. This is Tremella. This is mostly just a wood rotter. This is a jelly fungus. These fungi are mostly water. They're like 90-something% water. And they only show up during the rainy season to release spores. And then as soon as the weather dries up, these things wither to nothing and just disappear, essentially. This is usually not a pathogen. You'll almost never get questions about Tremella, but if somebody brings it in saying, hey, what's this? It's a jelly fungus. Okay, so those were some examples of basidiomycetes and what they do. The next group of fungi that we're going to talk about are Ascomycetes. And these are really important to us as plant pathologists because a whole bunch of plant pathogens are in this group. And we've seen them already, a whole bunch in, for instance, when you saw that orange that we looked at that had the little black things poking through, this is a picture of the side of one of those flask shapes that produces spores out of the surface of whatever it's infecting. So these Ascomycetes don't tend to form big structures like mushrooms. There's a couple exceptions to that. But most of these form these little tiny flasks inside the leaf or fruit tissue of whatever they've infected. So here's a picture of Annulohypoxylon. It grows on dying oak and woody tissues, and we slice through it. You can see these flasks in the side view. And this, once upon a time, each one of these was filled with spores. They've released the spores now, and now you just get these empty flasks built in the side. This is a morel. This is one of the ones that breaks the rule about mushrooms because a morel is an Ascomycete that produces a mushroom-like structure. But notice that it doesn't look like the classic Basidiomycete gill-capped mushroom. Instead, this has the same flasks, but just stuck on a stalk and made really big. So you can still tell this is an Ascomycete. At least every time you make a rule, there's some. Now I'm going to tell you there's a Basidiomycete called a false morel that looks like this, and you're just going to remember that these mushrooms just don't read very well, and they don't know the rules they're supposed to follow, and it's very inconvenient for those of us who like to make rules, but it works out. There's always something that breaks the rule. So this is a picture of Annulohypoxylon, which was this. We sliced through the side of this. This is what we were just looking at. We sliced one of these balls in half and then got up really close with a microscope. So this ball is just a whole bunch of those little flasks all sitting on the outside of oak bark. So this is an Ascomycete. Now we're going to miss some of the other stuff. I'll show you more Ascomycetes in a minute. Now these are the Zygomycetes. This Zygomycetes we're not really worried about from a plant pathogen perspective. You will see them sometimes. They're mostly refrigerator molds because Zygomycetes are the molds. So they do this kind of stuff. They're the stuff that grows in your refrigerator, forms the science experiments that make you go, ooh, you know, that kind of stuff. But these guys grow not inside the plant tissue. They tend to grow on the outside as well. So they just these big masses of mold that eat stuff. These are a problem for orchardists and folks like that, mostly in storage. They're usually not a big problem in the environment, although there's one, Botrytis, gray mold. It attacks roses. It attacks all kinds of things out there. It attacks peaches, and it is a Zygomycete, and it does this kind of stuff. And it can be a problem, but for the most part, we're not seeing many of those. And then we have chytrids. Now, the only reason I'm bringing this up, we do not have to worry about chytrids at all. Chytridiomycetes are generally just waterborne fungi. They're only a problem if you're trying to grow aquarium plants or aquarium other things. However, they are shaping our world. They are the thing that is causing the mass extinction of amphibians across North America. This was brought in from some other part of the world, and it infects the skin of frogs and salamanders, and it makes it impossible for them to osmoregulate, or impossible for them to hold water in and regulate their salts and things like that. It destroys the skin and kills the animals. So this is why yellow-legged frogs are now endangered for a large part of it. Why red-legged frogs are endangered. Most of the California native amphibians are struggling with this, and they may just disappear. And it's because of a fungus called the chytrids. We're not going to mess with the chytrids anymore beyond that. And then there's the oomycetes. Now, these are not true fungi any more than dolphins are fish or bats are birds. These share an entirely separate evolutionary pathway from true fungi, but they're more closely related to kelp. But you can think of them as kelp that's lost the ability to photosynthesize, or just found that it was easier to infect other plants than it was to photosynthesize themselves. So they're lumped in with fungi because they look like fungi and they act like fungi even if they aren't. So this is responsible for these are Phytophthora and Pythiums and one or two other, and downy mildews and a few other things along those lines. There's also a new group called Phytopythiums now. Not that it matters, but these are all plant killers in one form or another. And they're responsible, as we might remember, Irish potato famine was caused by Phytophthora in the beginning. It was the first plant pathogen ever truly classified as a plant pathogen. So we've known about this one for longer than any of the others, and it's not even a true fungus, and we're still struggling with it. And it's one of the things that causes sudden oak death, and it caused the Irish potato famine, and it's a big player on the world stage in terms of extinctions and famines and other fun things. And this is what it looks like. It looks like a fungus. It acts like a fungus. So things that we call fungi, they all have some things in common. They all have hyphae, these little tiny structures that are these little the filaments that grow through the soil and into things. They all have fruiting bodies that many of them are big and very recognizable. Others are not so much. And here's a great Ascomycete fruiting body. Remember how he said that they form their fruiting bodies inside of a structure? This is your classic Ascomycete. You've got no mushroom. It's just little rings of fruiting bodies being formed in the fruit. In this case, the fruit being the pea pod. And they all produce spores. So this is another fruiting body. This is a fungal fruiting body that's a Basidiomycete. It's a truffle of, this is a puffball technically. It's related to the truffles. And this is another fruiting body. This is a resupinate Basidiomycete. So that resupinate means to, like supine, means to lie down. So instead of forming a true mushroom, it just forms this thing, this gill, if you will, just a gill and nothing else. And all the Basidia are on that. And they all form spores. And the spores are a useful way of classifying them. So this is a Phytophthora with swimming zoospores that hunt in the water. These are Entomosporium. So that's a true fungus. It's a Ascomycete, and the spores look like insects. They got this passive way of diffusing through water, but they don't actually, though they look like insects, they don't actually move themselves. They just float passively in the water. And this is oh, come on. Anyway, it's one of the sick house fungi. It's really common, and its name is escaping me right now, and I'll remember it in 2 minutes after we got off the slide. But it's also, you'll notice the very characteristic spores that are 4 to 5 chambered, sometimes even 6. And I still can't remember the name. It's okay. So many fungal pathogens leave signs. This is Entomosporium. this infects all kinds of things in the rose family, and you'll see it on Toyin a lot. You get this beautiful red halo around the infection point. It turns black, and it's got this gummy oozing that shows up in the middle that often forms the leaf epidermis to crack, and the spores come oozing out in wet weather. This is powdery mildew. This again attacks, it's very fond of new growth of oaks and all kinds of other stuff, peas and roses, and it likes high humidity, cool temperatures. This is the perfect time of year to start seeing, we'll start seeing powdery mildew shortly. This is Ganoderma. It's a, these are all fungal fruiting bodies. They're always that fungi exist. This is Trametes, again, on citrus. This is turkey tail. It starts out as just something that gets in through old pruning wounds, and it digests wood, and then when it gets big and mean, it can start causing problems for the plant. This is Verticillium. This is an Ascomycete that grows internally on the structure, in the vasculature of the, I think this is a pear, if I recall correctly. Anyway, all of these are fungi. These are the kinds of structures you'll see from them. One of the problems we have when people bring stuff in is they'll bring stuff in that's pretty dead. And if it's old enough, there are plenty of fungi that break things down that didn't have anything to do with killing it. And if somebody brings you in a stump that looks like this and it's been dead for a year or more, and they say, oh, this fungus, it must be killing the plant. It's like, that plant's been dead for a long time, and I have no idea. You need fairly fresh symptoms to be able to tell the pathogens from the saprophyte. Saprophyte is something that lives on dead plants. So that's going to be a challenge that you're going to have. Some of the other challenges you're going to run across with, but is telling what caused it ultimately. So you'll get leaf and fruit infections. I think most of you guys know this stuff, right? Black spot on rose. So that's pretty straightforward, usually having to do with too much water on the leaves. Oh, here's another good one. This is Anthracnose. This is sycamore anthracnose. Anthracnose is a horrible term. You're going to hear it. I don't want you to pay much attention to it. Suffice it to say, anthracnose generally just means a leaf spot, but it's an ancient term used by plant pathologists, and they've been abusing with this it ever since. Technically, it just means something that has a very specific kind of asexual spore that it produces. I don't want to go into the whys and wherefores because it gets really confusing really quickly. All I want to say is, if somebody talks to you about anthracnose, just understand it's a fungal pathogen that usually causes leaf spots, although some of them make their way into the bark and other tissues. I hate the term. I wish we wouldn't use it. You'll see fruit infections. Again, things along this line, Aspergillus is what this was. That was the name of the that was the name of this fungus I couldn't remember before that we were seeing, and that's what's causing this as well. This is Verticillium. These are stem infections. They're clogging. Verticillium looks like this in rose, and it looks, wait, didn't we have one more? But it's a classic killer of Japanese maple, especially in Marin. It's clogging the vasculature down here, and that's why you will see it killing off part of the plant and not part of the other plant. You can see it in here. It's clogged the image on the right. The image on the left is what a healthy tissue should look like. That's all Aspergillus growing up through those vessels, plugging them up, not allowing the plant to move water through it and nutrients through it anymore. And then we have things like root infections. This is, again, our malaria on some orchard tree. I don't even know what this one is anymore. But you can, by the time you see these big clusters of mushrooms at the base, it's already been working on the tree at least for a year, if not more. And at this point, that tree's done for. There's nothing you can do to save it. What you need to work on doing is saving the trees around it by cutting this out, getting rid of the roots, much of the roots as you can, and then maybe doing some other stuff to protect the trees around it. Okay, so I feel like I've been talking for too long and you guys probably need a break. What do you think, Kay?
That's fine. How,
You know,
long, how much longer do you have on this presentation, Stephen?
A lot. How much am I already over time?
No, you're not over time. Why don't we take a 15-minute break?
should we ask a couple of questions that someone asked first? Just one question?
Absolutely.
Yeah, Jeff asked, Stephen, when you say some spores hunt, can that be equated to, say, a larval stage for the fungi?
Yeah, that's a pretty good way of thinking of it. The spores, yeah. And we don't talk about it in those terms, but and I've never even thought about it in those terms. But yeah, that's exactly what you could think of it as.
I was just trying to think of it as kind of like do the spores need to be in that hunting phase for a while before they start growing a new, like a new fully formed fungus?
No, generally speaking, they are happy to go very quickly. The spores are really a dispersal mechanism. So in some ways you can think of it like a larval stage, because it is a stage that well, a lot of times the fungus will spip the spore stage. You can get the sporangia, or the fungus will simply invade new tissue. The spores don't have to go straight into you know what? The analogy breaks down. No, they do not have to spend much time at all. Sometimes that spore stage will be incredibly brief. The fungus will just hop into a spore, infect another plant, and then boom, right back into the true fungal stage, if that makes sense.
Yes, thank you.
And then there was one question I lost way back, Stephen. Arlene asked, is cutting it out the only way to eliminate mistletoe?
No, you can wrap a tree. I'm not recommending this necessarily, but you can cut the mistletoe off without cutting it out. In other words, you don't have to remove the branch it was attached to, because you still have the haustoria of the mistletoe in the branch if you just cut the branch, if you cut the top of the mistletoe off. But then you have to wrap the branch in like multiple layers of heavy garbage bags and then seal the ends off with really well with duct tape or some other kind of that seals that section of plant in, so it cannot get light for years. And if you do that and it can't get light, the mistletoe will eventually die, and you don't have to cut it off. But it's challenging. The problem being, of course, that usually when you wrap something in garbage bags, you have something that still makes it inside there, like little sow bugs or roly-polies or earwigs or something like that, and they'll be scurrying around in there. And then you'll have things that crawl up on top in the things, birds, raccoons, that'll hear those sow bugs in there, and they're hungry, so they'll start pecking through the black plastic to try and get the sow bugs and other things out. And they'll destroy it, and so you'll have to go back up and you have to rewrap it every time it gets messed up. That's not an insignificant problem to try and do. There are no easy fixes for it. People have been working with, and I hear that some things like propiconazole may work reasonably well for killing powdery mildew. I'm sorry, I said that wrong. For killing mistletoe, but I have yet to see any really, that's all hearsay kind of stuff. I have yet to see good research done on things that you can spray on mistletoe that will kill it, that won't kill the host plant, because they're both plants. So, I mean, if it's an herbicide, it's an herbicide. So, you know, if you spray mistletoe with glyphosate, it'll work, but you'll also kill the host plant. So you got to find something that'll only kill mistletoe and not the host plant, and that's a real challenge. Propiconazole is technically a fungicide. It's also a plant growth regulator. It messes with things. So somebody told me recently that they're working on propiconazole as a potential treatment for it, but I haven't seen results yet for it.
And Stephen, the last two questions from Glenda and Arlene are similar. How do you know if a mushroom at the base of the tree is helpful or hurtful? And Arlene asked, does mushrooms growing under a tree always mean that the tree is diseased?
So the answer to the second is easier. And that is, no, most of the time mushrooms growing near the base of a tree are a good sign, because most mushrooms are beneficial to your tree, because the tree trades, so they're called mycorrhizal mushrooms, and the tree's trading with fungi underground because it's easy for the tree to get a couple of things: water, and it's easy for the tree to get sugar, because it takes the water and makes sugar out of carbon dioxide and sunlight, and there's plenty of carbon dioxide and there's usually enough sunlight. So the tree's good at making sugar. The fungi need the sugar. They're not so good at making sugar. They're good at breaking things down. So they will trade the tree for sugar for stuff like nitrogen and phosphorus that they can get out of dead bugs, dead leaves, and other things they find in the soil. So there's a trading relationship going on there. So most mushrooms are from the fungi that are beneficial to the tree. So just leave them alone. The only ones you're worried about are generally these clumping mushrooms that form right at the base. That looks like armillaria. So to answer the first question, the way you figure this out is you grab some mushrooms and then you go and hit David Aurora's book and see if you can figure out, and you make spore prints and you morphologically ID it and say, oh, wow, that's armillaria. That's a tree killer. The funny thing is, some of these things like armillaria that are tree killers aren't so bad if you eat them. Like, armillaria is not good eating, but it's not poisonous, and you can certainly make something out of it. It'll be a very bland, unremarkable whatever you make, and it can be kind of tough if the mushrooms are old, but you can eat armillaria. Don't go out and eat everything that forms a clumping mushroom at the base of a tree, because there are some things that they won't kill you, but you might be praying to the porcelain god if you get your ID wrong.
Could you give the name of the author of the book that you were mentioning?
Is David Aurora? Here, I've got it. It's Mushrooms Demystified by David Aurora.
Thank you.
And it's quite the tome, but it's really good if you want to get into that kind of stuff. He has pocket guides that are a little bit smaller. The frustrating thing for me is whenever I go use the pocket guide, I always get interested in something I've never seen before, and it's never in there because the pocket guides are the common mushrooms, right? So, and
why don't we take a
one of the
10-minute
great things
break?
about David Aurora is he's local. So if you've got questions, I think he's living in Santa Cruz right now. You can actually reach out to the mycological societies and get to know them. And that's the other thing to do. So you go down to with Kevin Sadlier at Green Genes. He's part of the Marin Mycological Society. He knows his way around mushrooms. So if you've got questions and you want to do this stuff, you can go work with him.
So Stephen, is it okay to take a break to 11:25?
Sure, yeah.
So we'll see you all back in a little over 10 minutes.
Sounds good.
Thanks, Susan.
Long way to go. I know. Okay. So more talking head. So the fun part is figuring out whether there's a pathogen or a saprophyte. So what we're going to see.
You're a superstar student. He knows everything.
We did this. Root infection. So your treatments for each of these different things is going to be fairly species specific. Some things like Verticillium, you're going to focus on plant health because the plant can recover from this and outgrow it if conditions are close to ideal. Other things, like some of these Ascomycetes that are rotters, once they're in the fruit, there's not much you can do for the fruit. So the leaves will get shed, the fruit will get shed, and the plant can fight it that way. For other things like armillaria, once the plant is been compromised by the armillaria at a certain point, there's not much that anybody can do for it besides remove the tree. I think I covered that already. Other things like some of these anthracnose diseases are pretty tough. They don't respond well to fungicides even if you did want to use them. So generally, and they don't cause enough damage that generally treatment's required. As long as you can keep the rest of the plant healthy, it'll drop the infected leaves and grow a new set. So there are a bunch of different terms that get used, and I want to sort of cover them a little bit. Now, these are, as opposed to classification terms that we talked about, Basidiomycete, Ascomycete, you know, fungi versus bacteria and all the other things. This more describes what's happening with a plant. So as I mentioned, anthracnose is an archaic term. It means that the plant is infected with a fungus, and the fungus is this group called Fungi and Perfecti. They're all asexual. So, and in this particular group, they're all Ascomycetes, I think, that are asexual. And they form this group, this little structure called Nasirvulus. You don't need to remember any of this stuff. The only reason I'm telling you this is that's the technical definition, and it has to do with the way it reproduces on plant material. And it usually shows up as a leaf spot on veins and twigs. And we don't need to worry about it. It's just a leaf spot. Anthracnoses are just not that different from anything else that produces leaf spots and that kind of thing. So you can ignore that one, although you will run across it. Cankers, on the other hand, is an actual useful term. This is a localized bark lesion. So a canker is sort of an infection in the bark, or sometimes in the wood tissue if it goes deep enough. So this is a canker that you're looking at here, caused by a Phytophthora infection, but there are a whole bunch of them. And there can be perennial cankers that sort of don't kill the plant very quickly, and the plant tries to grow around it, and the canker gets bigger and bigger and bigger as the plant grows and it struggles. And there are annual cankers where the plant can actually just stop it and heal over, and you'll see some of these things. We may get a look at some of these. A syndrome. Now, this is difficult to take a picture of or show you anything of. Sudden oak death is a syndrome, right? Most directly, it's a disease caused by a Phytophthora, but then the plant goes through a decline as a result of that disease, and then other things happen to that plant. Notably, for oaks anyway, in sudden oak death, there are beetles that hunt drought-stressed oaks, and the Phytophthora causes the plant, it cuts the top of the plant off from the bottom of the plant, making the top of the plant basically drought-stressed, which attracts the beetles, and the beetles are what do the rapid part, put the sudden in sudden oak death. So sudden oak death is a description of a syndrome, and the name was coined before we understood what was going on. You can't really take a picture of that very easily because it's a bunch of different things all going on at the same time to cause it to a plant to decline. So just so you know the difference between, say, a syndrome and another thing, a syndrome is a complicated thing that usually involves multiple pathogens or a pathogen and a pest, those kinds of things. And then wilts are what we were sort of looking at before. Verticillium is a classic wilt. Dutch elm disease is a classic wilt. Chestnut blight, I believe, is also pretty much a classic wilt. So these are some of the more severe things that attack whole populations of trees in particular. But Verticillium, as any of you who grow tomatoes knows, is also a big player in tomatoes and things along those lines. So it's not just trees. It can be problems, and it kills whole plants when it goes, because it's clogging up all the stuff that connects the leaves to the roots. And without the roots, the leaves can't survive, and without the leaves, the roots can't survive. Okay, so you're also going to hear some other terms that we throw around, and this wasn't saved, was it, properly? No, it's just, I had another version of this, damn it. Okay, well, I'm just going to have to put up with damping off is a rapid death of sprouts and seedlings from the soil up. So it's these little tiny sprouts that come up, and it's usually a greenhouse issue that's done in the soil tray liners, and you get the 2 little cotyledons, and the plant puts out its first little leaves, and then it just falls over. And those are damping off. And there's a number of different things that cause them. Fusarium and Pythium are 2 of the most common. Rhizoctonia is also up there. Blight is a rapid, generalized death of leaves. So chestnut blight is called that because it gets in through the leaves and then it affects the tissues lower down, and eventually usually gets to whole plants. There are some abiotic conditions that can be called blights because it causes a generalized rapid death of the leaves. So that's all it really means, is blight just means that. Galls. These are usually caused by insects, sometimes caused by fungi or other causes. Sometimes they're almost abiotically caused. Some plants produce big old, well, usually when they're abiotic, they're called burls, but these terms are all fairly fluid. It usually means you get this large woody tumor grown in the tissue. And sometimes that, like redwoods will do that just as a response to serious drought stress and a lot of sunlight on the bases of their trees. They'll build these big burls as an insurance policy. And all it is this mass of sprouts just ready to go. So if the top of the tree gets blown over, dies for any other reason, the plant will just sprout out with a whole bunch of new stuff. Other things, particularly there's a bacteria called Agrobacterium Tumefaciens that can cause galls that are actual tumors. The bacteria gets into the plant, injects its DNA into the plant DNA, and that's what taught us how to do genetic engineering and all that other good stuff. So there's a whole host of different galls, but they're usually all essentially tumors of one form or another. And then there are rusts. Now these are usually little orangish spots that show up on plants. Somebody brought it up when they looked at sunburn earlier and said maybe that's a rust, and some rusts can look a little bit like sunburn, just like that. Most of the time they show up as a lot of 1,000 little orange spots on an otherwise green leaf. But they don't have to be orange. There are yellow rusts, there are black rusts, and there are white rusts. But they're all fungal infections, they're all Basidiomycetes. And they generally infect leaves, but there are exceptions to that, sometimes stems. Okay, enough of this. So bacteria, these produce no signs. They usually produce fairly distinctive symptoms. So what we're looking at here is fire blight on pear, and you can tell it's fire blight without going through hardly any effort, because you can see it got in through the flower bud. So when you look at this big drooping thing, you can still see the little bracts on the bottoms of what used to be the flower, and you can still even see the stamen sticking out. And the fire blight usually gets in through the flowers. So this is how, and it kills the flowers off so fast that they don't have a chance to form incision zones. Those are the zones where, say, the leaf, I keep wanting to point at the pictures and you guys can't see that. The incision zones are where the leaves or fruits form a little scar tissue just before they fall off so that the plant doesn't have an open wound showing. And fire blight kills the tissues so fast that the plant has no chance to form incision zones. So all of the leaves are still dangling, and the flower that the disease got in through is still dangling from the plant. So this is one of the convenient things about this, is you can almost ID this. If you see something that's in a pome, so it's an apple or a pear or one of their close relatives that's still stuck on the tree and black or brown, and it didn't fall off, you got it. It's fire blight right there. Now, there are other blights that go in here. Bacterial blight is similar. It's usually caused by Pseudomonas. It works very much the same way, except it mostly infects stone fruits, not pomes. So the stones of the cherries, the plums, the peaches, the nectarines, and so forth. and it's caused by Pseudomonas. That said, Pseudomonas doesn't usually get in through the flowers. It usually gets in from frost damage. It's a frost damage specialist. It has little proteins on its coat that line up water molecules just perfectly so that as soon as the temperature hits 32 or even sometimes slightly above that, just the tiniest little bit of frost, and it'll just boom, build these huge ice crystals. And it uses those ice crystals to cause lesions in the plant cells by bursting plant cells. And that's how the Pseudomonas gets into the plant cells. So it has a different method of working, but in the end, it ends up looking fairly similar to fire blight, except that you don't usually have the flowers still attached. Okay, then other things that are caused by bacteria. Oh, and this is how you can tell fire blight anyway, at the right time of year. It's not always a good diagnostic tool because these symptoms are only there when the fire blight's still growing in the tissue and the plant's growing and they're fighting it out. So this red flecking you see, if you just take a scalpel or a very sharp knife and you just very shallowly dig into the bark, you'll see this brilliant, well, maybe not brilliant, but still bright red to oxblood color of tissue flecking through what had been green. This is all fire blight infection going on. And it tends to look, okay, so that's fire blight. Bacterial blight, tends to look a lot like frost damage because essentially it is. It's bacteria making frost damage more severe on spots. And this is why you'll often run across weird frost damage patterns. Like only 4 leaves got it out of your whole plant, but it's a whole hedge. Why did these 4 freeze? And people often say, oh, it can't be frost damage because then the whole hedge should have been affected. No, what you've got now is you've got Pseudomonas in there causing the frost damage to occur earlier than it otherwise would. That's how it makes its living. So it's, you know, whether you want to call it frost damage or you want to call it bacterial blight, it's sort of one and the same because the bacteria are making the frost damage more severe in order to get in there. Okay, so wetwood is yet another thing that you will probably run across. It's reasonably common. It's often called slime flux, sometimes called alcohol flux. It's caused by Erwinia, which is the same class of bacteria that causes fire blight, but it's not fire blight. It's in the wood. It's deep in the wood in this case. And what's happening is with a typical slime flux, it's getting into the bark, usually only in 1 or 2 tiny places, and it's kind of like a skin infection. It's not usually that bad for the tree. But it's getting in and it's digesting some of those sugars that it gets in the bark of the tree and turning them into poop, you know, gross, nasty, slimy stuff. So if you take this slime and you run it between your fingers, it's going to be slick. It's not going to be sticky. It's going to be brown and it's going to smell like dirty socks. Or sometimes if it's really fresh, it doesn't smell like anything. It'll be slick and not terribly brown and a little bit clear and not smell like much of anything. This is opposed to, say, exudates caused by Phytophthora, which is a different infection, which will be really sticky and they'll be usually quite red, and they'll often smell vaguely like a wine barrel. And that's typically the oozing you get from Phytophthora amorum and other things. And that's just pure defensive chemicals being put up by the tree, and it's got so much in it that's just oozing out. That's not wetwood. That's the thing we're contrasting wetwood to. But, and it's a sort of an indicator for sudden oak death. But wetwood is slimy, usually foul smelling, kind of slick, and not fatal to the tree. Not really an indicator that you even have anything going that terribly wrong. Now, I'll qualify that. With elms and poplars, sometimes we have bacterial wetwood go septic, and it can get through the whole tree, and it can take the tree out. That's rare, but it has happened. Then we get another version of slime flux called alcohol flux. It's essentially the same process, but the alcohol flux is gotten so deep into the tissues and there's so little oxygen available that it can't, and the plant's fighting back, and that it can't really digest all these sugars all the way down into, you know, sort of this slimy, poopy stuff. Instead, it's just got alcohol, so it's fermenting. That's how it's making its living. And this will be very sweet. It'll smell a little bit like juicy fruit gum. It'll attract yellow jackets and butterflies who will try and feed on this. Well, they're not just trying to feed on it, they're feeding on it. It is mildly alcoholic, and it doesn't even taste bad, if you care. And then the one other thing that is typically caused by bacteria are these huge galls. Well, they're not always huge, but they're caused by a disease called Agrobacterium Tumefaciens. And this is what taught us to do genetic engineering. So this bacteria gets into the wood. It snips off a piece of its own DNA into a little segment called a plasmid. It can exude it through the cell wall, and it puts it into the plant. And that DNA fuses into the plant's DNA and causes it to grow this tumor. And then the bacteria lives happily ever after inside the tumor. This is what taught us how to do genetic engineering. We used, for the beginning of genetic engineering, we used the Agrobacterium Tumefaciens little plasmid, that little snip of DNA, pulled out the part that the DNA that it wanted to key in, and we put in whatever DNA we wanted it to key in, and it would go in and put that DNA into the host DNA. And that's how you can genetically modify tomatoes or whatever else it is that you want to do. So, you know, nothing that, even our latest cutting edge technology, it's just copying nature again, which I think is kind of cool in its own way. I'm not saying genetic engineering necessarily is, it has its uses, but I think it's amazing that that we're still discovering stuff that makes your head explode. Okay, so bacteria, the treatment for these tends to be relatively pretty species specific when available. This was a treatment once upon a time. It was offered for your Felco pruners. You could put this little container, and every time you snipped your Felcos, it would squirt a little bit of antibiotic onto your tools to sterilize them. At one point, they were actually putting not just antibiotics. Well, yeah, it was antibiotics. It was true antibiotics. It was like streptomycin and stuff like that. So that when you cut, you sterilize the pruners between cuts so you couldn't transmit fire blight when you were pruning fire blight out, which is a big risk because bacteria are pretty good at you know, getting on infected tools, and then, you know, every other cut you make after that is infected, which is why we don't prune fire blight out in the spring unless you're disinfecting between every cut. Otherwise, you need to wait until mid to late summer when most of the bacteria have died off. Then you can cut with reasonable assurance that you're not spreading it to everything else, although you should still disinfect between every plant. Okay, so viruses. These rarely cause significant damage in landscape plants. They don't produce any signs. The transmission can be by a variety of different means. Sometimes it's by grafting or sucking insects. Sometimes it's by, sorry, I meant to say that differently. Sometimes it's by grafting. Sometimes it's by sucking insects. Sometimes, in rare cases, particularly with some things in the squash family, you can just walk by a plant and get it on your pants, and then you will transmit it to other things that have it. So that can be a problem. So try to, if you can figure out what the virus is that you're dealing with, there are tests available to do this kind of work in certain cases, generally not for the rarer viruses, but for the more common viruses that have big ag applications. They've come up with ELISA tests that you can use. They're fairly inexpensive, but they sell them in batches. So, you know, even though it only costs, you know, $6 to run a given ELISA test for a certain thing, you got to buy them in batches of 20. So that's $120 minimum to buy a batch of these things. So, you know, you got to have enough work to justify buying. Otherwise, if you're not using an ELISA test, diagnosis is almost primarily by symptoms, which is distortions or discolorations of leaves. And the other thing is, even if you figure out what it was, most of the time the only option you have for curing something that's infected with a virus is ripping the plant out. So, you know, before you go saying we want to test for this, my question to you is, if we come up with a positive on the virus, are you willing to rip the plant out? Because if you're not, what's the point of running the tests? Okay, some of the viruses don't cause big losses. There are some exceptions. This is black line and walnuts. It gets between the walnut cyan and the paradox rootstock if you're running a paradox rootstock, which is just a varietal of walnut rootstocks. It kills walnuts if it gets into them. So that's a problem for the walnut industry. Tobacco mosaic on tomatoes will basically wipe out your tomato harvest. So there are some things that make a big difference out there. Tomato ring spot on peach can cause losses. Apparently it suppresses peach production. Not that we have much. I have never diagnosed tomato ring spot on peach in Marin yet anyway. Tobacco streak on tomato is a problem. I see it every year, but it isn't enough of a problem. Your tomato plant will still keep producing reasonably well, even if you get tobacco streak on it. It comes in from weeds in the nightshade family. It's carried by insects. So we get questions every year. Maybe not this year, since we didn't hold the tomato market. We'll probably get a lot fewer people bringing their master gardener bought tomatoes in and saying, what's wrong with my plant? Phytoplasmas, I've only seen a couple, and they were only when I said I've never seen this stuff, and a professor said, hey, oh, I know of one, and we went out and looked at one tree that had peach yellows virus, or sorry, peach yellows. It's not a virus. Other than that, never seen it. Parasitic plants. We see this all the time, especially up around Sebastopols. Leafy mistletoe, dwarf mistletoe, dodder, bindweed, broomrape, they're all parasitic plants. We know the leafy mistletoes pretty well because they're easy to see. They're the classic thing that we see in a plant that looks like this. A dwarf mistletoe is only on conifers, and it only grows out when it wants to reproduce. Otherwise, it spends its whole life living inside the plant. So this is what a dwarf mistletoe looks like. Obviously, you don't usually get pine trees producing berries. So if you see a pine tree producing berries, that's what you're looking at, a dwarf mistletoe. This stuff is bad news. It can kill pines eventually. This is dodder. It wraps around. We have a number of native dodders. They're really prominent out in the salt flats around China Camp. You can see whole regions getting taken over by the native dodders. There are some exotic dodders that if CDFA finds that you've got them in your yard, they will come and tear everything out that's got it because they are seriously bad problems. So dodder, when it does get going on a tomato crop, looks a little bit like this, but it's not impossible to stop. Then we have things like bindweed, field bindweed. Now this apparently has some kind of parasitic function where it parasitizes plants below ground. I'm not exactly sure how that works, but it puts it into the same categories as broomrapes. So bindweeds are, if you don't stop them, and they can be really difficult to control, bindweeds can take over entire fields. And there's, these things go down 10 feet, and if you break them off as you're pulling them out, then they're just like dandelions. They just start all over again. And since they can parasitize roots, they can grow for ages underground before you ever see them above ground. So they can create really big problems for getting rid of. This is a broomrape. These are related to brooms, which is part of where they get their name, but they parasitize other plants, which is where they get the other half of their unpleasant name. So they are in the heath and heather family for the most part, but what they actually do, instead of parasitizing plants directly, they fool mycorrhizae into making them think that they're good trading partners, but the broomrapes take the sugar from the mycorrhizal fungus and they take the nutrients from the mycorrhizal fungus. So basically they're feeding off of they're thieves, and they're stealing from the supermarket that all the other plants trade with. And so the only time you ever see these things, they come up from the ground, they're not green, they're this tan color, and they produce flowers, and they go and the bees feed them, they produce fruit, and then they go right back to living underground the rest of their lives. They don't do any photosynthesis. They're just thieves. And the treatment for all of these things is to remove and try and prevent reestablishment. But of course, most of them live internal to plants or underground. So getting rid of them is challenging. Root nematodes. These result in plant wilting and stunting because they do a lot of damage to roots. What you're looking at in this picture is the nematode. The nematode is curved, and you can see it's got this little stylet, which looks like a mushroom growing internally to the nematode. And you can see the stylet just beginning to puncture the cell of a plant. And the plant cells are all at the top of the picture. So the nematode's coming in from the bottom and feeding, and you can see a little piece piercing the interior of that plant cell. And then it just sucks the nutrients out of the plant cell and in the feeding process. These guys can be really difficult to ID. It's not that hard to take a plant apart and figure out whether or not they have nematodes. The problem is most nematodes are actually beneficial. They help break down things in the soil, and just the plant parasitic nematodes are difficult. And the problem is IDing the nematodes and telling whether they're beneficial or not is challenging. It's not so hard if you have root knots or other clear indications of damage, but if you've just got nematodes in the soil, who knows whether they're brown. So this is what root knots look like in roots. Now, if this was a pea plant, you'd be stuck a little bit because peas also produce nodules that they put rhizobium into, which is how they fix nitrogen, and they need those nodules to survive. So peas can be a bit challenging, but pea nodules usually just look like these very tiniest little nodules that you can see up in the picture, these little tiny round things. The bigger nodules, peas almost never produce nodules that big. And the pea nodules, when you slice them open, will be red because they have hemoglobin on the inside, which is why they look a little bit like blood. These nodules, for root knot nematodes, no red on the inside at all. So some things to think about. I don't expect you to remember all this stuff. I'm just introducing you to the ideas. You can look all this stuff up on IPM online, or you can look up, how do I tell a pea nodule from a root nematode nodule on roots, and all that stuff's available. But the nematode ID, I can't do. In fact, even the nematologist at UC Riverside, Ole Becker, he doesn't do it anymore. He sends all of his stuff out to CDFA. There's one nematologist in California that does most of the ID, and it's expensive. It costs $250 a shot. So, yay. So we are done with our plant pathology lecture, at least for now. So the reason I ended with Thomas Malthus is just a reminder about why we care about plant pathology. You'll remember that Thomas, the Malthusian sort of dilemma that he brought up was that the populations increase exponentially, and food can be increase in food occurs linearly. And when you take a line and you intersect it with a curve, sooner or later, no matter what you do, you end up with a problem. We've been able to alter the line, jack it up higher and higher and higher for a while, but when we start hitting some numbers around 9 billion, we're going to start having a hard time feeding everybody, no matter what the heck we do. And so we're all going to start, this is going to be a big issue for the next couple of generations. So we've kept it at bay for now, but I don't know how much longer. So now we only have the sudden oak death thing to do, right, Susan? Right, Kay?
Right. And can we take a half-an-hour break for lunch before we?
Oh, absolutely. I'm tired of talking too. You guys are, I'm almost as tired of talking as you are sick of listening to me. So.
No way.
So how about we come back at 12:30?
Sounds good.
And everybody do a 7th inning stretch and get some food.
Sounds great. Thank you.
Thank you.
Guys
Thank you,
Great. Thank you.
Stephen, thank you.
Ready to go.
Look like I gotta take this off. There we go.
And Stephen, do you want people to ask you questions remotely by chat or just to interrupt you?
I'd prefer everybody interrupt me. It makes it feel a whole lot less formal.
Okay, does everybody hear that? Interrupt Stephen.
Okay, so without putting too fine a point on it, sudden oak death is, as we covered a little earlier, the name of a syndrome. But the causal agent, the thing that drives everything in this system, in this syndrome, is Phytophthora ramorum, which is one of those Phytophtheras we just finished talking about. It's a fungus-like organism, as much as bats are bird-like or dolphins are fish-like. This is fungus-like. It is not a true fungus. There are a lot of really important differences. But since it behaves a lot like a fungus and it looks like a fungus and all that stuff, we're still going to call it a fungus. We're just going to call it with a small f. It causes 2 diseases. One of the diseases epidemiologically is much more important, and that is it's a foliar blight. So it causes, oh, come on. Are you not working? Why has my screen stopped? I think I've got a technical issue on my end here. Give me a second. Everything's not working. Oh, there we go. Okay. Okay, it causes a foliar blight. This is all that sudden oak death looks like when it's behaving as it wants to behave. I know I'm anthropomorphizing horribly here, but so the Phytophthora ramorum is essentially a leaf spot pathogen. What it wants to do is get on leaves, eat a tiny chunk of the leaf, reproduce on that, and then get launched onto, have its kids launched onto another leaf so they can make their living. So this is what it looks like on bay laurel. It's not a huge problem. I mean, bay laurel doesn't like having its leaves infected, but it's not killing bay laurels by any stretch of the imagination. We used to not know where this thing came from. We've now found it in the jungles of North Vietnam. I'm talking northern Vietnam. I know North Vietnam is not a country, it's Vietnam, but northern Vietnam. It's loose up in that part of the world where it's fairly cool, and it is just causing little leaf spots like this. It's not killing everything in the environment. It's not killing a bunch of trees. It's just this little tiny leaf spot that's fairly nondescript. And that's all it wants to do. Unfortunately for us, this is a very good way of getting around in the world, if you will. Once it gets into a nursery or something like that, it forms a little leaf spot and nobody even notices it. Gets on rhododendrons, gets on pieris, gets on a whole bunch of different things, and that's how it spreads. The problem for us is when it gets into the wildlands in the United States and in Europe and other places where it's not native, the oaks there don't have any resistance to it. So but it's pretty much doing the same thing it's doing in South East Asia as it's doing in North Europe and Northern America. It's just hopping from leaf to leaf to leaf. The problem is when it gets on oaks, it's capable of penetrating the oak tissue, and it doesn't want to penetrate, it doesn't want to get into the oaks, but its option is do that or die, because so it goes into the oaks, and the oaks can't stop it, and it causes these huge lesions in the oaks, and then it kills the oaks. It doesn't get any benefit from killing the oaks other than it gets a few weeks longer to live, because it doesn't make it back out of the oak to have more to send its genes on. So that's a wildland issue for the United States, where it kills oak and tan oak and chinkapin and a few other things, and it's killed millions of oaks so far. So that's really the issue here. Please note, despite public perception, it does not spread from oak to oak to oak. It only spreads from a leaf host to a leaf host, and when it gets on an oak, it kills the oak, but it goes down with the ship. It's done. That doesn't mean it's okay to be moving oak firewood from one place to another. It still can be theoretically moved from place to place on oak firewood. It's just really hard to imagine how it's going to get back out into the world at large from there. And that's the big issue. So when it spreads, It's moving in wind-driven rain, just like we said it would before. Just like in Southeast Asia, it's just dripping from one plant, again, the wind picks it up and it moves to the next one. But when people move it, when they pick up infected plants, when they get it on their shoes and their car tires, when they run livestock through areas that are infected, they move it. And that gives it a new opportunity to infect new things in new places. So this is what it looks like on an infected tan oak. This one was taken in Big Sur. And you can see the bleeding is pretty dramatic. Now, if you stick your finger in that bleeding as opposed to bacterial wetwood, this is sticky and it will try and glue your fingers together. Now, of course, you can pull your fingers apart. It's not super glue. And it'll leave all these little tiny threads between your two fingers. And if it dries on your fingers, it gets to be a real bear to take off, because that's what it does. These are defensive chemicals made by the oaks, and they're made to bind proteins. So if you can imagine being a beetle and you've got proteins in your eyes, and you are boring through the tissue on this stuff is binding to the outside of the beetle's body and making life very, very difficult for them. Ordinarily, that's enough to stop them. But in the case, it's not enough to stop this fungus.
Stephen, question.
Yeah.
So are there some oak trees or types of oaks that are more resistant to it than others? Or
Absolutely.
ones that are more at risk than others?
Yeah. I will get into that. I'm going to address your question a little bit later in the talk, Ellen. So yes, great question, and we'll go to that in just a moment. Okay, so we deal with sudden oak death by growing it out in culture. We were trying to do that with these. I don't know if you can see this plate, but here it is. That's not sudden oak death, unfortunately. This is a negative plate. It's got everything else in the sun growing on it, except for sudden oak death. This is the same. Also, nothing, no sudden oak death on it. You can see what sudden oak death does look like in this plate in the slide. But we run these agar plates all the time to test for it. Here are, it produces a number of different spore types. So one spore that can germinate directly is this sporangia, and that can just, for Phytophthora morum, that can break off, land on a leaf, and it will directly grow a hyphae, and the hyphae will go and attack that leaf, and off it goes. If it lands in a puddle of water, it will release these zoospores. The zoospores are excellent swimmers, and they have antennae, and they will hunt for susceptible hosts in the water. It can also produce these chlamydospores, which are, without making too fine a point on it, essentially seeds, if you will. They've got this really tough coat. They can survive dry, warm temperatures. They can survive dry environments. And they will just wait until conditions are moist and cool, and then they will hatch out and release zoospores or hyphae or whatever. So, impacts on the state, we've killed well over 1 million trees at this point. It's killed well over 1 million trees at this point, and there are at least another 1 million, much more than that actually, currently infected. This talk, I put this together at the end of 2018. It's now the beginning of 2020. So a year and a half later, we realized that these numbers are pretty low. It's infected more than 2 million acres of forest, which is less than 10% of the high-risk forest in the state of California. So the damage is still going to continue to go on. It affects the ecology in the sense that forests look and act differently, and there are wildlife impacts. For instance, the dying tan oaks you see in the image in front of you are the only major acorn producers in redwood forests. And there are a number of species, including jays and a number of different squirrels and chickarees and things like that, that all depend on acorn crops in order to survive in a redwood forest, because it's one of the larger, more nutritious nuts available. And when the tan oaks all disappear out of these forests, what's going to happen to the animals that depend on them? It's going to change the ecology of our redwood forests, at the very least, probably a lot of other forests as well. There are safety impacts in that hazard trees die. Some of them will fall over much earlier than people expect to. We can get into exactly why that is. But there are hazard tree dangers. There are fire dangers withstanding, as you can imagine, having those tan oaks stand there in that forest doesn't make that forest any more fire safe than it was before. In fact, it puts it the other way. There are economic impacts in the case of the cost of implementing quarantines, the costs of tree removals, the costs of firefighting efforts when you've got additional fuels in the forests now. And there are emotional impacts. And I know this sounds kind of wishy-washy, but let's take that as emotional impacts and put them into raw dollars. In Marin, the median property value, at least a year ago or so, was $1 million. So we're going to use just your median house instead of picking on anything extravagant. When you have a large mature oak in front of your house, that can be worth up to potentially 10% of your property value, much more typically around 5% of your value. So just to keep from being kind of crazy with things, let's just take 5%. It could be as low as 2%. Let's take 5% as a median value in there on a median house. On $1 million property, that's a $50,000 oak tree. That's what people are willing to pay in addition to whatever they would pay for a regular oak, for a regular property, for a property that has a really nice, magnificent oak in the front yard. It's about a $50,000 difference. That's what I'm talking about, emotional impact. People care, and they're willing to put money where their mouth is for this stuff. And even if you don't care a whit about trees, maybe you only care about money. I know that's not any of you. You're all master gardeners. But even if that's all you care about, if you, then $50,000 is still something you care about. Okay, so impact. With fire, you're going to hear a lot of conflicting information about sudden oak death. That's because, unfortunately, some people have been apparently misquoted. Other people have stated things that were taken out of context. So I just want to clarify one thing. When you look at the great big picture, the really big picture, sudden oak death has no impact on fire. That's because if you test, I don't want to go too far into this, but if you test a given point on a property and say, does this property have sudden oak death? If it's got the very beginnings of the infection, all it's got is those little leaf spots. And yes, the property is infected with sudden oak death, but it hasn't changed anything on the property. And that's in the very early stages. In the very late stages, sudden oak death has been there for 20 years. It's killed most of the trees it's going to kill. Those trees have been removed, or they've fallen, or they're on the ground. And really, there's no difference in the fire behavior once the susceptible trees are down and on the ground. It doesn't really change how fire behaves. It's only in the middle stages. When it's killed some big trees, they're still standing, like what we see in this picture. That's on a steep slope where you've got standing fuels. That's where sudden oak death makes a difference in fire. The number of plots that have sudden oak death that are in this condition are relatively few. And there are a lot of other things that are killing trees in our forests in California, including beetle attacks and drought and all kinds of other things. And so when you look at it overall, sudden oak death has no net impact on fire behavior in infected forests versus other uninfected forests. It does cause standing dead fuels, and that's not really so much a disease issue as it's a fuels management issue, because other things cause these things too. And if we just focus on the sudden oak death, we're missing the boat. The point is, if you want to control forest fires in your forests, you need to focus on standing dead fuels, and it doesn't matter what killed them. So while you hear a number of different things, all I'm trying to say is the presence of sudden oak death or the absence of it from a given forest is an academic point. The only thing we really care about is whether you've got decent fuel loadings or you've got fuel loadings that are out of control for your site. That's what we need to worry about. Okay, so we're going to talk a little bit about timeline, and because it's really hard to understand how this disease moves if you don't get a chance to if you don't understand how it moves in time. So, I wish I could turn that off. Let's take a look. In 2005, we had a wet spring. There was mortality associated with that doesn't occur for tan oaks for about a year, and mortality associated for coast live oak doesn't start for almost 2 years, a year and a half to 2 years after that initial wet spring. So when people first see this wet spring, they don't see any dying trees as a result because it doesn't happen for a while afterwards. The association isn't necessarily there. In 2006, we had another wet spring, and we had some mortality associated with those. In 2007, we had a dry spring, so very little sudden oak death spread in 2007. In fact, probably next to no spread occurred. But in 2005, 2006, all the other trees were infected, and that's when they actually began dying, is around the winter of 2006 and into 2007. That's when you started seeing it. And the other thing that contributes to this is, remember, Phytophthora is infecting all those connective tissues that conduct water up and sugars down. And that means that the canopies are cut off from water because the roots are starving and because the vessels are plugged. So the real stress is when a dry year hits for these trees because they're getting less and less water. So the dry spring was the real peak. That's where you really saw your mortality occurring. So people see dryness and they see the mortality occurring, and it seems like, oh, they're getting dry. It's a drought issue. It's not. Well, I mean, it's related to the drought. It's also related to the rainfall. It's related to all these different factors. The other thing that happens in dry springs is beetles like the ambrosia beetle that hunt for drought-stressed oaks are out looking for drought-stressed oaks in dry weather. And lo and behold, we have all these trees that have had their root systems compromised, and they see these oaks, and they can see these oaks like you and I can see ripe bananas versus unripe bananas from 3 aisles away in the supermarket. We can tell just looking at that banana table which ones we're going to be interested in, which ones we're not. And then the beetles home in on because they see in the infrared spectrum, so they home in on the drought-stressed trees, and then they sniff them and see if they smell like they're good enough, and if they are, then they attack those. So we'll get back to the beetles and that kind of thing. The point here is that mortality rates increase following wet springs, and they drop off after successive dry springs, but the actual trees die in the dry summers. So what we're looking at is the aggregate mortality from 2005, 2006, really getting a start in 2007, running through 2008, and kind of continuing through 2009, and then they kind of go away. Because we had dry springs for a number of years after that, all the way into 2009. We had, in 2010, we had, 2010, we had a normal rainfall, cool spring with some rain. And in 2011, we got a really wet spring, and it started a whole new wave of mortality that really doesn't start showing up until 2012. So that's how it exists over time. Now, the other question is, how does it behave over space? So what this graph is showing you is, if we take the vertical axis here where it says spore count, if that is our host tree, that's a tan oak, a bay laurel. It could be a tan oak too. Let's take it as a bay laurel right now. If you are right underneath the bay laurel, the spore count is incredibly high. If you are far away from the bay laurel, the spore count is low. The spore count under normal circumstance is the red line that you see here. And you can see, and it's a logarithmic scale across the bottom. So 200 yards, 1.5 mile, 3 miles. The next thing would be like, 30 miles. So if you're right underneath the tan oak, you're getting really high numbers. If you're about 30 to 50 feet away, you're still at some risk. But once you're more than about 50 feet away from that tan oak, you're at almost no risk under normal circumstances. However, in the spring, we get these big wind events, and wind-driven rain events during storms. And that's where this dotted blue line comes into play. So the red line represents your risk if you're an oak near that infected bay laurel. The blue line represents your risk if you're a bay laurel compared to that other bay laurel of catching the disease. And what happens is, you'll notice that sometimes in wind-driven rain, you can get these things that go out 3 miles from your tree. So this means that just in wind-driven rains, just a few spores are getting spread as much as 3 miles, and they're not infecting oaks, they're just infecting other bay laurels out there. But when that other bay laurel becomes infected, then all the oaks around that bay laurel are now looking at this red line in terms of their mortality risk. And this is why it shows up in spots across the landscape and it isn't like some smooth wave. It's just popping up here and there, and then those spots sort of grow and coalesce as time goes on, as more bay laurels next to that original spot get infected and transmitted on. And then as oaks near all those different bay laurels all die. The problem here for tan oak is that it not only gets the disease, but it's a host. The disease, Phytophthora morum, can infect tan oak leaves and rains it down. That means tan oak is always right in this, at the very center of this, at the vertical axis on this. It's always in a high spore load count, which accounts for why it dies so much more quickly than regular oaks do. It's because it sporulates on tan oak's own leaves. So it's always there. This epidemiology I've described to you is true for Marin and Sonoma and most of California. As we start going north, the role of bay laurel doesn't count as much. And this is not due to a number of different factors. One of them is genetics. As we move into Oregon and Washington, bay laurel is not as good a host, and we can't tell you exactly why. We can just tell you that it is. So bay laurel no longer drives this once we're in Southern Oregon. The main driver of mortality is tan oak and rhododendron, not bay laurel. Okay, so this is a list of all the susceptible species that have been out there. Now, understand, when they say susceptible species, they are talking about species that can get it and you could get potentially spores off of. However, you'll notice that we have most of these in just white and a few of them are in blue. Let's see, no, it's not the right one. So these are the ones that really play a role that we've been talking about in this forest epidemiology. California bay laurel does not get it on its trunk. It does not die from the disease, but it's an excellent transmitter of the spores. Camellia also transmits spores to a number of to other plants around it. It's not as effective as California bay laurel, mostly because when camellia leaves get infected, the plant drops them. So they're not fluttering around in the wind up there spraying spores everywhere. Camellias are mostly, as soon as they become infected, they drop to the ground. They're just not transmitting very well. Although they're still producing lots of spores, they're all on the ground. They're not up in the air. Coast live oak is not producing any spores to speak of. It is mostly just a target, but it dies. So that's why it's listed here. Kalmia is good at, it's a nursery crop. It's not even in the California wildlands, but it's very good at moving this stuff through nurseries. So that's a problem. That's why it's on this list. Rhododendron is also really good at moving this stuff. It's not as tall as bay laurel, so it's not in the wind as much, so it doesn't send spores as far, but it also is very good at spreading the things. And it also, some cultivars of rhododendron, and there are like thousands of different cultivars of this plant. It's one of the broadest plants in the world. It's also one of the most highly traded. The reason rhododendron's here is because it's probably how it got here on the first place. In fact, it was a rhododendron nursery that it was traced back to as the original point of introduction. So we're pretty sure that's how it got here. And it's good at spreading the disease. Not as good as, it can't spread it over the same area that California bay laurel and tan oak can do it, but it's very good in its localized area. And it does, some varieties die from the disease. And then tan oak, I think we've already covered. It's both dying and it's also very good, like California bay laurel, at spreading it. And it's also large, so it's very effective at spreading those spores. So those are why those are all the hosts. The host list is pretty far out of date. This one dates as 2008, so well, you know, it's 12 years ago. The thing, and it has expanded since then. The thing is that all of the key players had been identified by 2008. Now we're adding a few other things that end up being a problem. For instance, yew, although yew's on here, in England is a big player. It's not a player in the United States for some reason. It has to do, we think, with both the weather in England and the varietals of yew we have here. I thought I heard a question. Nope. Okay, good. So new additions to the host list. Some of the ones that we care about that are here in Marin that are not on that list you saw from 2008. So this is Trostania, as I know it. It's been renamed Lophostomen because somebody has to get their PhD, adding more, more syllables, longer names, PHs. You got to do all that stuff because it makes your thesis much more valuable. These were taken in downtown Sausalito. This was found by Juan Ochoa at Bartlett Tree Experts, working with Suzanne Latham. This is not the typical dieback pattern we see. It hits just about every leaf on it, but the twigs survive and they sprout new leaves for it. Camphor. This was also found by Bartlett working with CDFA. This has been killing a number of trees all the way up into the Terra Linda area and even some in Novato. So this causes a slow decline, and camphor, unlike most of the other things, develops the disease on its twigs. So that's why you see whole patches of the canopy dying back, and it's sprouting from below where those twigs are infected, and that's why it's behaving the way it does here. So here's a picture of the camphor. It's a little hard to see, but if you trace some of the twigs back, you'll see, particularly in the middle of the image, you can see that it got it. You can see a little black spot, pretty much dead center of the image. You can see how it's infecting the leaves in these orangish puffy areas on the sides of them. You can see once it gets into the tip of the twig, it's dying back. Here's a better shot of the blackened lesions on the bottom of a camphor twig. This is what is killing the camphor trees here. 9 new species have been added since 2015. Some of these have been done by Bartlett, some just by CDFA, and some by other people submitting them. And again, it looks a little bit like camphor. You're getting the twigs dying back. That is what remorum means. Remorum is to branch. It comes from, words like ramifications. They come from their Latin roots. To ramify, to branch, consequences, those kinds of things. Remorum was a name for its ability to infect twigs. The big effects in California are the nursery contagion issue. Nurseries are really hard places to control diseases in. They tend to be fairly dirty. I encourage you to quarantine any plants you buy from any nursery, no matter how reputable, for a couple of weeks to make sure they perform reasonably well in their pots before you plant them out on your property. I know lots of people who've brought in some very nasty Phytophthora and other diseases onto their property from infected nursery stock. I'm not trying to paint the nurseries black or red here. I am saying that with diseases like Phytophthora that can live in soils or can be plants can be cryptically infected for years before they show symptoms, the nurseries can do the best job in the world, and it's still really hard to manage these things. It is simply the nature of the business. So it behooves you, I think it's caveat emptor at this point, it behooves you to take the precautions that are needed to protect your property so you don't have to wrestle with these things, because once you get a Phytophthora like this, you've got it forever. Anyway, okay, so we already covered the fact that the loss of tan oaks and redwood forests are going to have some changes to simply the way, even if we don't do anything else, the chittering of the chickarees and the squirrels and the calls of the jays are going to get fewer and farther between. Redwood forests are going to become quieter places, and unnaturally quiet. True oaks, the big things are slower to show symptoms than tan oaks are, and they are slower to die. That's that cryptic infection period, and you can have a tree that's that's really got the disease and from the outside does not appear to have the disease for some time. There were some questions a little bit about resistance, and then the questions were relating to not just within one oak species, but between oak species. So we're going to get to that in a minute. But there are good signs of resistance in the oak population. So, you know, I don't quite, if you're looking at it from the 10,000 year time frame, everything's going to be just fine. That is small consolation if you own a pocket of oaks on your 40 acres, all of whom are susceptible, because oaks being wind pollinated do tend to be fairly related to their closest neighbors, but the relatedness tends to go away as the farther away you get. You could get a cluster of trees on your 40 acres of which every single tree is susceptible, and there's not much you can do about it. Likewise, I know there are pockets of trees in Nevada where every single tree is resistant. And the we have some numbers to throw at this, and so I'm going to throw them out. The numbers are best guesses, really, at this point. Nobody has done a truly systematic survey because nobody has developed something that really tells you whether an oak is resistant or not that's commercially available. The problem with that is that there are a couple people, that notably Anna Conrad at The Ohio State University and Bryce McPherson working at UC Berkeley have come up with a test where they can test your bark extractives and be able to make a pretty good determination of whether your tree is susceptible or resistant to sudden oak death. Mostly it's whether it's super susceptible and pretty much guaranteed to die or not. That's how resistant it is. It doesn't really tell you. It just tells you that it's not on the very susceptible end of things. The problem with that is they've been telling us for 10 years they're going to get a commercially available testing kit made, but the market doesn't work out. When we start looking at price points, and maybe I just won't bother you with this, it's not commercially viable, at least according to the folks that say this is how much it would cost to develop it, this is what our odds of getting a return, and this is how long it's going to take. Most people say, I'd rather invest in something else. So, as yet, although the scientific methodology has been developed, there is no commercially available test for your oaks. That said, about when you talk to Anna Conrad and Bryce, they estimate that the resistance in the susceptible population is about 17% of the population. So that's roughly a little less than 1 in 5 oaks is so susceptible that if it gets the disease, it's guaranteed to die pretty much no matter what. That disease is going to run through it. Those oaks tend to, like I said, develop in clusters. And if it's anything like a bell curve, we think we see at least that much resistance on the other side. So at least 1 in 5 oaks is so resistant that it probably never gets a disease. Or if it does, it gets the disease, it kills it and stops it, and it's done. I'll talk a little bit more about some of the research I've done on this in a minute and where some numbers come from. But this is what the big picture looks like. In areas where you have trees, and these are all tan oaks, this is Salmon Creek up in Sonoma County, not far from us. This is what it looked like shortly after sudden oak death went through. This had all been solid canopy before that. And this whole section of it was tan oaks, and all of them are dead to the last one. Tan oaks, the most quote unquote resistance we've seen is about 3%. And that population, it doesn't mean it's immune. It just means the disease develops much more slowly in that 3%. But 3% is such a small genetic bottleneck that even if tan oaks were to survive on that 3% somehow, and we don't think they even are, we think ultimately they're dying anyway. But even if you did have that, it's such a small genetic bottleneck that they'd all be very closely, all the survivors would be very closely related and therefore very susceptible to extinction from the next thing that comes along. Okay, so this is true oaks. This is a picture taken in Marin County out toward Tamaulipas. These were some of the large oaks out on the Gale Ranch. I believe this oak is dead now. But that's what a bleeding coast live oak that's got the infection looks like. So I'm going to talk just a little bit about native oak biology, because when we start talking about this oak susceptibility question, it helps to understand how oaks are related to one another and how they're grouped, and then it starts making a lot more sense. So oaks are linchpin species of our forests. That means that most oaks have, if you go out and visit them, you know, in fact, I hate walking with people and they're like, can you find anything wrong with my trees? And it's like, how many days do we have to do this? Because oaks have 1,000 things all quote unquote wrong with them. They've got 1,000 diseases, they've got 1,000 pests, and that's okay. They are linchpin species for our forest. There are 1,000 things that depend on them. So of course they're going to have 1,000 things feeding on them. And that's part of what makes them so central to everything. There are lots of things feeding on them, and there are lots of predators out there eating the things that eat oaks. So whole ecosystem chains depend on them. That's what we mean by a linchpin species. And oaks aren't passive in this process. They actively manage pest populations. We think of trees as being these things that just sit there and suck up water and photosynthesize. They're doing a lot more than that. Oaks selectively allocate defensive chemicals from one branch to some certain branches and selectively short other branches' defensive chemicals. Why? Can you think of what an oak purposefully, now granted, I'm more promorphizing here. Why would an oak short one of its branches' defensive chemistries? Can anybody think of a reason?
To make them more concentrated in the other parts of the tree that are healthier?
Yeah, what does it get out of doing that, Jeff? You're right.
It increases the odds of the whole organism surviving rather than trying to protect every little twig and branch from getting killed or hurt.
Good. So Jeff's on the right track here. So let's think about this from the species of a bird. You're visiting an oak. The oak has oak moths. Oak moths eat oak leaves. Now, if you're an oak moth, where do you want to be in the tree? You want to be on the sweetest, juiciest leaves you can find that don't cause you an upset stomach when you eat them, right? So if you're oak moths, you cluster on the leaves with on that branch that has the least number of defensive chemicals. Does that make sense?
Mm-hmm.
Why would that be an advantage to a bird? Because if you're a bird now, you don't have to hunt the whole tree down. You just have to find that one branch that has those low levels of defensive chemicals, and you can find all the caterpillars all on that branch. Maybe not all of them, but most of them. It makes the bird's life a lot easier. So the oak is helping the birds by differentially allocating its defensive chemistries. So it makes the bird's life easier on eating the caterpillars. So, I mean, I know we think of oaks as we don't tend to think of oaks as complicated, complex species that are active players in their environments, but they absolutely are. They do the same thing underground with mycorrhizal organisms. If it's short on one thing, it'll preferentially trade with the mycorrhizal fungi that can provide it with those things. So it's playing with the species that feed on it and it trades with constantly. So the reason I'm bringing this up is oaks differ in their ability to do these things, partly because they differ among each other and where they're adapted to being, and partly just within even a single species. Just like people, some are better at trading than other people, and others are better at making than other people's, and things like that. So let's go move on from here. Oaks are long-lived generalists as a group. Changes occur over years or even decades. That is hard for some people have a hard time wrapping their heads around that. They're like, oh, you know, I did some root pruning on my oak. Yeah, that was 5 years ago. It couldn't be affecting the oak now. No, 5 years ago is exactly when you should start expecting to see the results of the damage you did back then, because the effects on an oak's timescale occur really slowly over the span of years. So they're long-lived generalists as a group. They're promiscuous specialists as species. They get around a lot because they're wind pollinated. They hybridize. They do not follow the rules I'm going to set out here and tell you these are blue oaks and these are white oaks and these are California valley oaks and these are Oregon white oaks. None of those, the oaks don't care about those boundaries I just listed off. As far as they're concerned, they're all oaks, and you'll run across hybrids between species all the time, and that breaks the species concept. It doesn't really work that well with oaks. So that said, let's talk about the white oaks. Oaks only hybridize within their sections. So all the oaks I'm going to list here can hybridize with one another because they're closely enough related to one another that their children are viable. So Quercus lobata. This is a classic valley oak. It's one of the largest oaks in California. It's one of the largest oaks in the world. These things can reach well over 100 feet tall and sometimes over 150 feet wide. They have a lot of names. They're called California swamp oak, California white oak, valley oak, just white oak, post oak, a lot of different names. That's why we stick with Latin, because Quercus lobata is the only name, Latin name for this. Quercus douglasii. This is commonly known as blue oak. As you can imagine, you can see why. You'll notice the leaves are not nearly as deeply lobed as lobata, and they've got this, they're a lot darker, and they've got this powdery color that gives them a blue sheen at a distance. These two, so Quercus lobata, the valley oak, likes valley bottoms. It can handle water. It likes being near rivers and having a lot of things. Blue oak is a hillside species, pretty restricted. It hardly occurs at all in Marin and Sonoma counties. Only in the very easternmost part. It's really a Sierra foothill things. It likes the Central Valley foothills, really like. It does not like summer water. And yet these things are so closely related, their kids can be something in between. Quercus garryana, Oregon white oak. This tends to occur in shaded north-facing forest slopes among other trees. It looks just like Quercus lobata for the most part. The leaves are really similar. From a distance, you'd have a hard time telling them apart, except Quercus garryana is a little smaller, and it can handle shallower, thinner soils, can handle a little more drought, but it wants more shade. These trees are all native. They're all the leaves have these rounded tips on them. If I go back, look at the leaf tips on the lobes. They're round here. This hardly has lobes, but they are round where they exist. These lobes are rounded. So they're all lobe-leaved, and they're all deciduous. None of them are evergreen, except some scrub oaks. So Quercus dorata, believe it or not, is a shrub, evergreen, and it's closely enough related to Lobata, douglasii, and garryana that you can get hybrids between these and the other ones, which messes everybody up and makes oak taxonomy a nightmare, because you think you got it and you go, and it still catches me out sometimes, because you'll notice these have spines on the ends of their leaves. So some of the hybrids from this with Quercus lobata will have pointy leaf edges, and be evergreen. So they look like coast live oak, which is not even remotely closely related to, which is crazy. The other thing that we care about is every one of these is functionally immune to sudden oak death. Every one of them. So when I say functionally immune, I'm saying that, yes, we have been able to inject these trees with Phytophthora morum. If you cut out a section of bark and you jam a whole bunch of mycelium in there, they will get the disease. Yet somehow we have never run across one that's naturally infected, which suggests there's something about that. And it's thought to be the bark growth patterns on these. They don't expand their bark until later in the season, when most of the risk of Phytophthora infection is gone. So we believe it has to do with the growth pattern of the white oaks, that they simply never contracted. Okay, so that was the white oaks. That's one clade. They all hybridize. They're all immune. These are the red or sometimes black oaks. They get both names, unfortunately. These all get sudden oak death, and these all hybridize with one another. Coast live oak is perhaps the best known because it's probably the most common oak here in Marin and Sonoma counties. Again, because it hybridizes, you're going to see some interesting questions come along about exactly what's what. Quercus callogii is black oak. This tends to show up in forested settings or in the north slopes of mountains or in other shaded locations. It occupies some of the same places that the Oregon white oak would have been in the last one. And interior live oak. This is pretty rare in Marin County. It does occur in a few places on the eastern part of the county, but mostly this is California. This is a Southern California oak. This is a Central Valley oak, a Sierra foothills oak. And then Quercus parvula. This occurs around Mount Tam and some other places. This is a fairly rare oak, and they're still, if you want to start an argument among plant taxonomists, just ask them whether Quercus parvula is really Quercus whistlezennii or not. And that will, you won't hear them talk about anything else for hours. But this is considered by many people to be its own separate species. It also gets sudden oak death. The funny thing is, Quercus whistlezennii, this interior live oak, has never actually been found with the disease more than maybe once or twice. In other words, it doesn't appear to ever really get it. Even though we've tested it over and over again, it certainly gets it when you give it to it in the lab, and it is related to all these other species. So the jury's still out on whether this should be considered susceptible or not. That's another good argument starter. And then there's all these hybrids. So this is a picture I took of a chase oak. This is from Helen Putnam Park in Sonoma County, right next to Petaluma. And this is a hybrid between black oak and coast live oak. And it looks halfway in between either one. In mild winter years, it functions as an evergreen. If it gets to be a really cold year and we get a little bit of snowfall, it'll drop all of its leaves. It doesn't know what it wants to do. But it's pretty cool. And they're kind of rare, so it's interesting. But I've also seen these in Marin County. They're present at Alompali. I don't know how to, Alompali, however you say it. But notice that all these are either evergreen or have pointed lobes. There are no trees in here that are deciduous with pointed lobes. They all get sudden oak death, potentially, except we've never really seen interior live oak get it to any appreciable extent. Now, within that, some of these oaks, although they get sudden oak death, are really resistant. What that means is the tree will get the disease will progress to a certain extent before the tree figures out, oh, I've got an infection. The tree marshals its defenses and it kills the disease. And we know this because we've been able to test certain trees as positive. We've gotten the positives back. We grew them out in the lab. I've done it myself. And then we go back a year later to check on that tree and see how it's doing and whether it's dead or not. And the tree has completely stopped the infection. It's grown callus tissue over the old infection wounds and it's closing the wounds over. So there was a master gardener, Lad Sievenpiper, that was part of this program for years. I think he retired back in around 2011, 2012. But he had a number of trees on his property that we pulled sudden oak death out of, and which are still standing today, and they've got the scars to prove they had the sudden oak death, but they've completely healed themselves and cured. So these trees are probably on where we were talking about the other side of that curve, where the trees are pretty resistant to infection. So there are some trees that are so susceptible, they're pretty much guaranteed to die no matter what you do. That's the work done by, and that's about 20% of the population. If we figure that like this is a normal curve, like most distributions in nature, human height, you know, a number of other things, if you look at the distribution of anything, generally you end up with a bell-shaped curve. So we would estimate that roughly 20% of the population is so resistant that it's never going to get the disease to any appreciable disease extent in the first place. So that leaves about almost half of the oaks as being pretty much, you're wasting your time with trying to do anything for them, because some of that half are so resistant that they're going to die no matter what you do, and some of them are so, now I said that backwards, some of those trees are so susceptible they're pretty much going to die no matter what you do, and some of those trees are so resistant they're never going to get the disease in the first place. That still leaves us with another half or so of the population that's in this middle ground. They can't really, that you don't really know whether they're going to make it or not, and those are the trees where agrophos probably makes some kind of a difference. Now, all agrophos does, and has ever been shown to do, is reduce the rate of spread in the trees. Now that has been interpreted by some, Matteo notably, as suggesting that the trees may have enough time to recover, especially in light of the fact that some trees we know do recover. So that's been interpreted as meaning that we can make some of these trees actually healthy enough to survive indefinitely with repeated applications, or maybe indefinitely if they just get enough repeated exposures to the disease without dying, they develop what's called induced resistance. This happens with a number of different trees. A good example is Monterey pines and pine pitch canker. If they get the disease over and over and over again, over the span of about 10 years, they develop resistance. It's induced. They eventually figure out how to make themselves not get the disease anymore. They get immunity to the disease effectively. But if they get the disease and they die before that 10 years is up, then they're dead. So they never develop induced resistance. That's probably a factor in here. Nobody really knows in sudden oak death. So agrophos is one way we may be able to get the trees to survive long enough to develop induced resistance or something along those lines. This is a phosphate.
Does that mean it acts a bit like a vaccine?
The agrophos does not act like a vaccine. Agrophos acts more like an antibiotic, where it's going to slow the disease down enough or allow you to get past that disease, that disease enough times where you have a chance of developing induced resistance on your own in order for it to be more like a vaccine. So it's a great question. I'm having a hard time answering it because human immune systems or animal immune systems and tree immune systems are so different that analogies really don't, or similes don't work incredibly well. All I can really say is one thing that agrophos has been shown to do is stimulate certain metabolic pathways that increase the defensive chemical production within the tree. As animals, we don't really produce defensive chemicals, but we make use of trees' defensive chemicals all the time. Turpentine is terpenes, which are a defensive chemical made by pines. Tannins are defensive chemicals made by oaks. We use them for tanning leather. These things bind to proteins, lock them out. Turpentine is something you would never in a million years want to drink because it's really nasty. It's poison. So all these defensive chemistries are used to keep pathogens at bay. And so phosphonate ramps up the production of defensive chemicals in the oaks. That is not without consequences. That's expensive stuff. You're basically sort of forcing an oak to run a fever, if you will. Running a fever is a huge metabolic investment. And one of the reasons there's a big variety in the responses of oaks is thought that some trees have historically sort of gone down the evolutionary pathway of, I'm not investing a whole bunch in these expensive defensive chemicals. I'm going to invest those resources in foliage and fruit, and I'm going to out-reproduce the pathogens that come to me. Others have really gone down the defensive chemistry pathway, and they pay a metabolic cost for that because those chemistries are really expensive to produce. It's a little bit, it's going to sound really hokey, and I apologize for my dumb analogy, but it's a little bit like somebody living in a bad neighborhood making a choice between whether they're going to buy Top Ramen or a Smith & Wesson. A Smith & Wesson might keep them safer, might not, but it's really expensive to buy. Whereas you could buy a lot of Top Ramen for that Smith & Wesson. So which one is going to be your priority? Are you going to defend yourself? Or are you going to let the crooks come in and take what little Top Ramen you have? I don't know. Anyway. Agrophos is pushed by some. I'm not sure it's a gamble at best. Fully half the trees that you're going to probably potentially treat will either, it's a waste of money for one reason or another. And the other half, you don't really know whether it's going to boost your tree's defensive response enough to make it go the rest of the way. But it has been pretty much shown to work in some cases. So the question is, how much does it cost? And how much money do you have? And was it worth it to you to risk that money on preserving your oaks? The other question is, do you inject it or do you spray it on? Injections are preferred by some applicators because they get more of this stuff into a tree in a shorter period of time. So if it's going to have an effect, you're going to get a better bang for your buck out of this. The problem with it is it injures the tree, and because of that, you can't do repeated applications of this over and over again. Whereas the spray, you could put new sprays on this 2-3 times a year, no problem. And you're not going to hurt the tree by doing any of that. And eventually you'll get the stuff in, and it takes less time. Sprays are much faster, so you're paying less per application. So there's, horses for courses, whatever you want to do. Injection, surface application is lower dosage. You get a lot less into the tree, but it's very simple to apply, and it can be done multiple times a year. These are only really workable for specimen trees. You will read in the Marin IJ, there's a guy out there who's advocating for going out and spraying every tree in the Marin forests with agrifos. I think it's probably a huge waste of money since we know it's not even going to work on half of them. And then on the other ones, it's got questionable efficacy. But it might make a difference. In the grand scheme of things, since the oaks have a pretty sizable resistance within their population anyway, whether we do that or not isn't going to make a whit of difference in the long run. 10,000 years from now, nobody's going to be saying, man, I'm glad they did or didn't spray those trees with agrifos. It's going to make no difference. You'll hear different questions about application timing. This changes over time as we learn more. They tell you that it's about spring and fall, and that's not untrue. The truth is that the tree will only take this stuff up if it's actively transpiring. What does a tree need to be actively transpiring? Anybody? What's a plant need before it transpires?
Leaves.
Yeah, okay, so leaves, great start. We tell you to apply this in the spring and fall. If you apply this in the fall to a black oak, is it going to take any of it up? It's got no leaves. And the answer is no. It won't take it up because it has to be actively transpiring to do it. So if you've got a black oak, the only option is spraying after it's leaved out. Or we'll get into it in a little bit else. What else do you have to actively be transpiring? You got to have leaves. You got to have water, right? So if you've got water, and the other thing you really need is it's got to be warm enough and have enough sunlight to actually be moving the stuff through. So if it's kind of cool and rainy and it's only 48 degrees outside, are you going to take up any of this agrifos? And the answer is no. It won't even take it up if it's 50 degrees and cloudy. You need it to be warm, like above 60 degrees, sunny, and the soil has to be wet. The only reason they're recommending spring and fall as treatment times is because those are the only time of year in California where it's routinely warm and sunny and the soil's moist. In the summer, it's typically often warm and sunny, maybe not in some parts if you live in, but the soil is usually pretty dry in the summer. And in the winter, it's so cold and rainy, nothing's happening in terms of transpiration. The tree's just not pulling that much up. So that's why they recommend spring and fall. You can apply this stuff, you can apply agrifos in the middle of summer if you do a root drench 2 days ahead of time and just get a lot of water in there. The tree will be transpiring like mad right after that because it's sunny and warm. So then you can apply your agrifos in the middle of summer. If somehow you can manage to make, if you happen to be able to be psychic and you can time your things for, what was it? It was February this year. We had some 70-degree days right after it had rained. If you had applied agrifos then, it would have been taken up beautifully then too. That's all I'm trying to say. Okay, one other thing. Trees age, as they increase in size, they become more and more and more susceptible. That seems to be the case. Trees below about 4 inches in diameter are functionally immune, and as they get above about 4 inches in diameter, they start becoming more and more susceptible. So don't even bother treating trees less than 4 inches in diameter. And trees, really big trees, you can apply a lot to, and it may make a difference for you. Okay, so this is what I was basically talking to you about before, about bell curves and susceptibility. So she estimated, Anna Conrad and Bryce McPherson estimated 16, 17% at the top end in terms of plants that were highly susceptible to the disease, highly resistant to the disease, I'm sorry. They were able to measure, that's what they were looking at, was resistance. We estimate that depending upon the numbers you use, Ted Swicki and I have both done studies, and we're estimating the susceptible portion of the population to be somewhere around 40%. And that has a lot to do with the definitions of resistance and susceptibility. And I'm not going to spend a lot of time. Suffice it to say, Anna and Bryce defined things a little bit differently than we did when they did their studies. And so I think we're probably not disagreeing with each other very much. What we're really sort of saying is we're defining things a little bit differently at the start of our experiments. Suffice it to say, some trees are resistant and some trees are susceptible at the either end of the spectrum. The idea here is if you have and I've already sort of covered this, if you have a highly susceptible species and you apply agrifos, agrifos moves you toward the resistant end of the spectrum. But if you're taking a super susceptible tree and you move it a little ways and it's still susceptible, you've made zero difference. Similarly, if you take a tree that's already pretty resistant and you've just moved it more solidly into the resistant end of things, again, you've probably made very little difference. It's the trees in the middle that are probably on the edge of being susceptible. If you can move them into the fairly resistant end of things, that's where you're probably going to see the bang for the buck. And that's where most of the trees are. That's the big portion of the curve. So that's why we think there may still be some utility to agrifos. Okay, so what does, or agrifos is just one trade name. It's just the same thing as phosphonate. It's the same thing as reliant. It's the same thing as control phos and a bunch of other trade names. It's all phosphonate or phosphonic acid or potassium phosphonate. Unfortunately, a lot of different names for the same thing. I'm not a chemist. I can't explain to you why that would be. But the idea is that the phosphonate treatments help bolster the tree's natural defenses. If you've got a weak tree, it's going to have a weak effect. This is much better as a prophylactic. In other words, when the tree's healthy and still has a robust and defensive system, you can get a good bang for it. If you wait till the tree is already infected and the tree's already on its knees and it's already weak, you're not going to get as good a response. So it generally doesn't work as a therapeutic or curative treatment. And it appears to be essentially useless on infected tan oaks. And this is a picture of a tan oak that I had treated with agrifos the summer before this picture was taken. And then I went out to check on my trees in my experimental plots. And that's the tag and the whole 9 yards. And you can see the points where it had been bleeding and the rain had already begun washing some of the bleeding off. But the tree is functionally dead at this point, and it's only 6 months after I had started treating it. And that's because it was already infected at the time I had started treating it. I just couldn't see it. Okay, so, you know, agrifos isn't the only option. Many oaks are already resistant, depending upon exactly, you know, how you define resistance. Continual reinfection from nearby by bay laurels may mean that they eventually succumb even if they are resistant. Removal of bays may help those oaks survive. So here's some work done by Jana Valikovic up in Oregon. I did another study in SoCal demonstration before this was done, before Jana's work was done with the Garbalotta lab. I didn't take as good of pictures as she did though, so I'm going to use her pictures. So what this basically means is, remember that I said that bay laurel thinning, that if you were farther than about 50 feet from a bay laurel, it probably meant you were reasonably safe from the infection rates because you had to have fairly high you had to have fairly high spore numbers to cause the infection. That was that spatial graph I showed you. So the idea here is if you cut down all the little bay laurels right next to oaks, maybe the oaks have a better chance of surviving. So here is the before picture where we have some trees that were before thinning, and we simply took a bunch of the potential foliar hosts out near the oaks, and then we looked at whether the oaks survived. And lo and behold, we did have higher survival rates where the incidence of foliar hosts was lower. This isn't cheap to do. It's not that easy to do on a homeowner's landscape, partly because most people don't own acres and acres and acres of land in the first place. And this is sort of something that has to be done over the space of acres to be effective. Also, because if you do it over the space of acres, you're talking about teams of guys with chainsaws and chippers out there for days. It's not cheap, but it does seem to work. So we've had some people try it, and it is effective. There's some alternative treatments. Folks talking about forest decline claims. They're saying that we get acid rain in California, which is BS. Well, no, it's not entirely BS. All rain is acidic. So the pH of rain is about 6.5. And if you define anything below pH 7 as acidic, then we get acid rain in California. But when they talk about acid rain, they're talking about acid rain like they got on the East Coast, where the pH of the rain was 3 or 4, which is 1,000 times more acidic than pH 7. So I can't say that they're lying when we say we get acid rain, but plants are fine with pH 6.5 rainfall. They always have been. It is technically slightly acidic. That's not a problem. It's when you start messing with it and making it 10 or 100 times more acidic than pH 6.5 that we've got a problem. So people have been claiming that the forests on the west coast of the United States are declining as a result of acid rain. That's not true if we're defining acid rain to be something of around pH 3 or 4. The pH of rainfall hasn't changed on the west coast in forever. We've been monitoring it for over 100 years. It has not changed meaningfully. They've claimed that it's a soil acidification process. Soil acidification is a natural part of a maturing forest. Big mature forests live on acid soils. That's simply the way it has always been. Now, there is some truth to the fact that California, at least, was routinely burned, and that did inject some high pH stuff, which tended to correct that acidification thing over time. I'm not saying everything about these alternative treatments is false. I'm saying that they're extrapolating into areas that they've got no scientific credible proof for their claims. So if you want to go out and apply gypsum, or you want to apply other forms of calcium like oyster shell, and you want to lime your trees, the trunks, because you think it'll help them, you're not going to hurt anything. Go ahead, that's not a problem. There's no data to suggest it works. There's plenty of data to suggest it's not effective. These are pictures of trees that were limed by Lee Klinger. They're in West Marin right now. They're dead. They died as a result of sudden oak death that got them even when they'd been limed. There is every possibility these trees were already infected before Lee even painted the lime on them. I believe lime might be a reasonably effective deterrent to Phytophthora landing on it, but it doesn't stop the trees from getting infected. You'll hear claims for bark scribing. Bartlett has been doing this for a while. My predecessor, Pavel, how much time do I have, Kay? Or Susan?
How much time do you need?
I'm almost near the end. I'm just trying to, I've got a few more slides, and I want to know how quickly I need to move through this, because I don't necessarily want to bore everybody with my talks and my bark scribing studies if I'm
how about 10 more minutes and then questions?
Sorry, 10 minutes and questions?
Yeah.
Okay, so I'm going to skip over my bark scribing studies here really quickly because I don't think that's that important. Suffice it to say, bark scribing doesn't work either. Oaks are really good at stopping the disease all by themselves. I can explain to you what was done to how I proved that with the study I did, but we don't need to. We can discuss this if you want to come into my office or you want to talk afterwards. Call me up, that kind of thing. I'm happy to. Let's talk about hazard trees, because this is much more important on a day-to-day basis. A hazard is defined as something that has a target. A tree that's diseased and going to fall over in the forest is not a hazard to most people because they're not in the forest with the tree. if this same tree is over your driveway, over your garage, or over your kid's bedroom, it's a hazard, because if it falls, there's a target. So one of the questions is, how do I assess my tree to figure out if it's a hazard or not? Because if it potentially has sudden oak death. Here's one of the things to look for. This is Annulohypoxylon. I already showed it to you once before. This degrades the sapwood of the tree. The sapwood is the most important wood for the structural integrity of the tree. And by the time you see these things on the outside of your tree's trunk, this has already been digesting your tree for at least a year. If you see this on the outside of the trunk of your tree, this big supporting wood, if you see it on a little minor branch, don't worry about it. The little minor branches die and rot off of these trees all the time. But if this is on the trunk of your tree and it's near your house or your driveway or your egress, if you're trying to get out in an emergency, you need to have that tree seriously looked at, because this is a sign that your tree is not structurally sound. Even if it looks great on the top and it's green and full and healthy looking, you need to have it looked at. So this is a sapwood decay agent. It's part of the sudden oak death syndrome. Ambrosia beetle builds these galleries. Remember those dark black galleries you saw in the tree that had snapped off? This is what it looks like from the outside. This is all sawdust. This is because the beetles are not eating the wood that they're tunneling. They're just chewing it up and spitting it out the outside of the tree. So it's super fine. It feels like talcum powder. It's very soft and smooth between your fingers. And it coats the outside of the tree or collects in the crevices. And this means the beetles are busily building a nursery inside your tree, and your tree is at high risk for falling over, because those nurseries, not only the drilled holes weaken the tree, but those beetles are lining those holes with this ambrosia fungus that is actively digesting the tree from the inside. These trees can and do fall over when they're green looking, and this is one of the things climbers will look at before they climb the tree, because we've had people get killed trying to take down sudden oak death trees and climbing these things, and the tree goes, all it takes is putting a climber in it and the whole tree goes over with them in the tree. So those are the two things to look for. If you see those, worry. Okay, next slide. It's not advancing. How do I do this? Here. Okay, these are native organisms. They're not something that we're ever going to be able to control. Annulohypoxylon has been around for longer than people have. Ambrosia beetles have been far longer than people have. So we're not trying to control either one of these things. They're an important functional part of California ecosystems. You just need to know that these are the warning signs that your tree is going to go over, because the interior is going to look like this. You can see a little bit of hypoxylon hiding in that bottom. On the very bottom, slightly to the right, you'll see annulohypoxylon and the beetle galleries in this. This tree was guaranteed to go, and it did. Okay, so what about if I have an infected tree? What do I do about disposing of it? There is a quarantine in place. You may not move this stuff outside of the county, so you can't sell it to firewood people who are going to send it up to Yosemite, and there's a reason for that. We don't want infected material moving into Yosemite. Even if it's really hard for it to get out of the wood, it's still possible. So we encourage, if you have a dying tree, keep it on your property. Now, I understand some of us don't live on 5 acres where we can just stack a bunch of dead wood out on the back. So if it has to get moved, you can have somebody haul the stuff away. They will simply take it to a disposal facility. But it's best to simply leave it on site, because then you're not moving the stuff and having to pay for it. You could wrap the cut wood in clear plastic if it's got beetles emerging from it. The beetles are native. You could do that. It might help some of your surrounding trees, but the bottom line is these beetles are flying around every year anyway, so don't bother wrapping your trees in plastic necessarily. The best thing you can do for your trees, if you don't want them to get the beetles, is make sure they have water during the summer. They don't want a lot. Oaks hate water, but if we haven't gotten rain since April and it's now September, you could water them just once or twice during the summer, enough to give them a little bit of water to work with. That will let them not appear so bright on the beetle's infrared visual radar, if you will, and they will not attack your tree. Composting does kill the pathogens. So if you get infected material and you want to move it off of site, send it to your green waste bin, send it to the compost facility. I did the work on that personally. I wrote the paper on it, so I can promise you it works. Landfills and compost facilities within Marin and Sonoma are in compliance. They know what they have to do. They know they have to tarp their loads. They know that when you bring stuff in, it has to reach 131 degrees Fahrenheit. They handle it appropriately. Okay, next slide. So if you're going to remember anything out of this whole presentation, remember that oak mortality follows wet springs. There's a 1 to 3 year symptom lag. It does not move from oak to oak to oak. It moves from bay laurel to bay laurel to bay laurel, and it gets on oaks in the process and sometimes kills them. Agrifos is a preventative. The efficacy is not assured. It's a little bit of a gamble. Laurel removal works, but it's incredibly expensive, and it has to be done over a large acreages in order to really be effective. Hazard oaks, look for beetles and hypoxylon. Consider removal if you see them. Call a consulting arborist to have them check it out. If you're talking about disposal, keep it on site if you can. That's the safest thing. If you can't, you can have it hauled and composted. If you're looking for diagnosis, ask yourself these questions. Does it really look like sudden oak death? Is it on the full known host list? You can go to www.suddenoakdeath.org and you can find out what the known host list is. What are we talking about? Look, testing a foliar host, testing a branch host, testing a terminal host like an oak. Is it even in the infected area? If you're talking about Placerville, it has been tested to death. We've gotten some positives out of it. The infection has never survived. If it is in an infected area, and Placerville is not considered an infected area because it can't survive there, then yes, call it sudden oak death. If it's not an infected area and you still think it is, call the ag commissioner or somebody else and get it tested. And this is what you're looking for. This is how we test it on bay laurel leaves. It's far easier, it's far more reliable, it's far cheaper to do these kinds of tests. That's what it looks like on camphor. That's not that hard to get. This is the distribution of it in our area. All the red little triangles are sudden oak death positives. All the greens are sudden oak death negatives. All these sites have been tested. They're all part of Mateo's Sod Blitz program. Sod Blitz is a great way to get this stuff done for free if you want to participate in it. The link is right there in the berkeley.edu in the middle of the left-hand side of this slide. He is going to be conducting the Sod Blitz this year, so you can sign up to go do that. I think his first one is in April sometime, coming up here for Marin County. These are all resources for you to get information on. The most important one up here is the Sudden Oak Death website, www.suddenoakdeath.org. The sod map is up here. That's Garbalato's thing. That's linked to his Sod Blitz. If you're interested in the fire things, the work done by Margaret Metz, which are the next two listings, get into some really the nitty-gritty of fire and sudden oak death and how they do or do not really interact. Maya Bay, which is the fifth citation down, is talking a little bit about some of the more meta aspects of fire and disease. And then if you want to talk about CAL FIRE clearances, because you're concerned about fire, I've included that on this as well, because a lot of people were concerned about that. And then I'm happy to talk to you about bark scribing more if you guys want to talk about that. But actually, we're out of time. So we'll just I've got some slides on that if anybody cares.
Stephen, I have a question about the state parks and national parks and the spread in the parks and what it's doing to our park system.
Okay.
So what is it doing to our park system?
Not anything that it isn't doing to the rest of our lands. It's increasing our costs on maintenance on our park systems. That's for sure, because they now have one more thing that's killing trees in the parks, and that's one more set of fuels to manage. There is no way to treat sudden oak death in the parks in Marin and Sonoma counties, because it was the horse was already left that barn by the time we identified what the problem was, and that was in 2000. It was already too late to quarantine it in California by the time we even identified. It was remember at the beginning, sudden oak death, we didn't even know this pathogen existed. So maybe this is too cavalier, but the parks is what's going to do to our parks is what it's going to do to our parks. And it's not likely to change our parks in any appreciable way except that in the redwood portions of our parks, we are likely to see some flora and fauna changes. In the oak woodland sections of our parks, we're likely to see bay laurel continue to expand at the expense of oaks for the next couple hundred years. And then if fire gets brought back in, that should correct an awful lot of that whole thing, because it should eliminate bay laurel from, so if we look at the, bay laurel's been expanding. If you look at pictures, even from the 1930s and 40s, from the first aerial photographs, if you look at where we have a lot more forest than we used to. And the majority of that forest is in non-oak. It's in Douglas fir and bay laurel and a few other fairly aggressive, I don't want to call them invasive because this is their home after all. You can't invade your own home. But Douglas fir and bay laurel were often excluded from a lot of these areas because repeated fire kept them out. Now that we've reduced fire to next to nil, they're what's expanding, and the increase in bay laurel is sort of pushing the oaks back. We think we're already seeing a lot of the most susceptible oaks eliminated, and the oaks are gonna, the bay laurels are not gonna keep pushing the oaks back for long. And if fire gets reintroduced, it will probably reestablish a status quo. So the question becomes a little bit of one of how well can you predict the future? How much fire is going to get reintroduced to our parklands, and what are people comfortable living with? And I don't know a lot of the rest of this stuff, I'm sorry to say.
Are there any other questions?
Yes, I have a question. Will there, over many, many years, will there be enough oak trees that became resistant, and that will become our forest, and the trees that didn't develop that resistance, will have died off, and so we'll just have a forest of resistant oak trees?
Right. That's why I was saying 10,000 years from now, everything's going to be fine. We think that process is already underway. In fact, in some of the areas that were originally hit, we're seeing oaks come back, and they don't seem to be developing necessarily the same rate of infections that they did. Now, that's a little hard to say, because remember, anything smaller than 4 inches is functionally immune anyway. So, and it takes oaks a few years to get to 4 inches just. So we don't really know. We just think that things are already moving in that direction.
But not in our lifetime.
It's a little hard to say. Yeah, possibly we'll start seeing a measurable effect within our lifetime, but a measurable effect is often not a visible effect. You know what I mean? We might be able to say in in the next 20 years, somebody may be able to sit down and say, we've done the bark analysis that was pioneered by McPherson and Conrad, and say, look, it's expensive. We tested these trees, and out of this small population, they have way higher levels of the extractives that suggest they're resistant than the previous population did. So we're thinking maybe it will. I don't know. I know that Bryce and a couple other people and Ted Zwicky think that we're seeing lower infection rates in the older, more infected forests. But I think that's just people guessing. They've also said a lot of stuff that hasn't been true. So I don't know. I don't think scientists have any real solid, when they're guessing, I think honestly, their guesses are as good as anybody else's. The only difference between them and anybody else is they can test their guesses and tell which ones aren't true.
I have 2 questions.
Yeah.
One, the bay laurel are there 2 different trees? Is there the bay laurel and the California bay laurel? Or is that the same tree?
That's the same tree. It's Umbellularia californica, okay. And people shorten California bay laurel to just bay half the time for shorthand. The problem with terms like bay is there's a European bay or even bay laurel, so it gets messy. So it's just a really long common name. I wish there was a better one.
Okay, and also, are they considering changing the name of sudden oak death? Because it's sort of a misnomer, isn't it? Because it's not really sudden.
We've been saying that for a long time. We've been ruining that. It's not sudden. It doesn't just affect oaks, and it doesn't always kill them when it does. They tried. they tried calling it Remorum blight for a long time to refer to the foliar infection process of it, and just reserve sudden oak death for when it was actually killing oaks. And it just doesn't stick. Sudden oak death is a name that people remember and they hang on to, and people like talking about it. And Remorum blight, you know, when you talk about Remorum blight, people say, what's Remorum blight? And you say sudden oak death, and they're like, oh, okay, got it. You know, and you're back to where you started from.
Stephen, Tanya Rhodes and I have the same question. Should host plants be removed from areas around oaks? Tanya just transplanted 3 rhododendrons to an area very close to her oak tree, and I've got oaks surrounded by camellias and rhododendrons. So what is your suggestion?
You know, this gets hard because on personal landscapes, those plants are there to serve a purpose, and that purpose is to keep you happy. So let me ask you this. What would you lose? What would you rather lose? Would you rather lose the camellias, and which are beautiful plants, and keep the oak, or would you rather lose the oak and keep the camellias? Because that really is the ultimate choice you're going to end up having to make. For me, I like camellias and I like rhododendrons, but I like oaks more. And it's a lot easier to put a camellia or a rhododendron someplace else. And it's really hard to put an oak back in that's going to be anything of that size in your lifetime. So, and when you look at the simple raw finances of it, an oak makes a much bigger bang for your buck in terms of your sale price of a house. At the end of the day, camellias and rhodies simply don't, not to the same degree. So to me, I'm keeping the oak, and in fact, the camellia that's growing in front of my house, everybody loves it. It is just loaded with flowers. It's gorgeous. And I'm gonna cut the freaking thing down. And my mother-in-law is horrified, but it's going, because it's close to the oaks that I wanna keep. And it's also right underneath the eaves of the house. It's a fire hazard. I don't know who put it there. It's a terrible place. I'll put another camellia in someplace else.
I had a question.
Please.
Are people noticing that there's a specific species of tree taking over the now opened up areas where the oaks have died?
I don't know the answer to that, Glenda. That's a great question. Most of the time when people do talk about trees taking over, they're talking about Douglas fir. So I think the answer to your question, I don't know the answer to your question, but I think the answer to your question is yes. It tends to be fairly weedy species like Douglas fir. Now, Douglas fir is a California native, so it's not really fair to call it weedy. But any plant that isn't the plant that you want, I mean, the definition of a weed is a plant out of place, and out of place is a human construct, certainly not a natural one. So yeah, I think most of the time when people are talking about what comes back when you lose oaks, they're talking about the things they're afraid are coming back. They're not talking about the good things that are coming back sometimes. So I don't hear a lot of talk about rhodies. I don't hear a lot of talk about other oaks coming back, even though they probably are. What I hear people talking about are Douglas fir and Scotch broom and French broom and gorse and a few other things. And those are, most of those, aside from Douglas fir, are weeds. And I do know some people who have active management projects to keep the Douglas fir down because they're worried it's going to wipe out the oak recruitment in those areas.
Okay, thank you.
For what it's worth.
You mentioned that this can be spread by car tires and any number of other things coming from nurseries. So it's hard to imagine if you bring in something from a nursery that's simply quarantining for 2 weeks, will tell you whether, not necessarily a carrier, but just, some of these things have survived on the plants or leaves themselves. So how else do you prevent, especially for under oak plantings, how would you prevent bringing this in from a nursery?
Emery, you're absolutely right about that. And when I said 2 weeks, you caught me. Really, it has to be 6 weeks. That's what Dave Rizzo recommends. And I apologize. You do need to quarantine them for much longer than that. And the reason for that is this. The nurseries are using stuff, they're using fungicides on a fairly regular basis. And one of these is Subdue Max, which is Mefanoxum. And it takes about 6 weeks for the effects of Subdue Max to wear off. Now, this is sold as a fungicide that's supposed to kill Phytophthora, but what we've been able to prove is that most Phytophthora have developed resistance to it now. So now it's functioning as a fungistat. It keeps the plants from developing symptoms. So the nurseries think they're doing a great job. But as soon as the fungistats are removed, the Phytophthora get right back to work. They are not dead. And that's what the 6 weeks is for, is basically it takes a while for those fungistats to be purged out of the system, and long enough for disease symptoms to start to show up, that the plants that are growing out will start to show symptoms. And if they start showing symptoms by the end of your 6 weeks, you've got a problem and you need to not plant them out in your landscape. So thank you for that.
Any other questions?
Sounds like we're good to go. I hope we stayed kind of on time, guys.
Good enough. Thank you very much, Stephen. We really appreciate you being with us today and being with us through Zoom, and feel honored to have you.
Well, the feeling is mutual. I am only sorry for all of you wonderful Master Gardeners, because this looks like a great class, and I would have so really enjoyed speaking with you guys in person and, you know, hanging out with you guys and talking about the other things you guys do. So I'm looking forward to, if and when all the things get lifted, seeing you guys all on the desk and getting a chance to at least meet you there, because it's going to be good.
And we'll invite you to graduation wherever and whenever it is.
Yay. Okay. Well, thank you. Thank you for that. And have fun, you guys, with the rest of the class. I know it's hard, but